Thanks, John. Thanks for organizing us, as always. Nice job. So John asked me if I would introduce Fred, and I said absolutely. So in case you guys don't know me, I'm Sandra. So I have worked with Fred. We were talking this afternoon. I think it's been 7 years that he was my boss. So I do know him. Fred has been involved in CDISC since the beginning, basically. So he knows a little bit about what he's going to talk about today. He's got, you know, some nice background. You talk about send, right? So yeah, not today. Usually, yeah. Not today, but usually. Okay. So yeah, so Fred knows of what he speaks, and we should all listen to him and learn a lot. He's been a really good guy to work for, and he's really informative, and yeah, I think you're going to enjoy. I'm glad that he made it out here today. He is not a Bay Area person. He had to take a plane to come here and present today, so awesome job. Thanks, Fred, for coming out and joining us. Welcome.
Thank you, Sandra. How are we doing on the webinar, Bernard? Bernard?
Oh, yes.
Can you hear me?
I can, but you're a little faint.
Okay.
There we go.
Where you're going to actually be talking.
Can you hear me now?
Yes.
Okay. Guess we're good to go, right? Okay.
Wait. Okay. We're almost good to go.
Almost good.
Sorry. Let's show Fred.
Terrific.
Okay. Start from the beginning, and I think we are good to go now.
Okay. I'm going to talk about what's new in version 3.3 of the SDTMIG, and also version 1.7 of the SDTM. How many people here are like SDTM people? Okay, well, that's half the audience. How many people have actually looked at version 3.3 of the SDTM implementation guide? How many people think they're eventually going to look at version 3.3? Okay, so most companies now, version 3.2 came out in 2013. And probably most companies, if they're kind of moving in a future direction, would have implemented that 2013 version of the SDTM implementation guide. But for those who want to keep up with everything, now we have version 3.3. And what we're going to talk about today is what's happened in the SDTM in the meanwhile between these two versions in the 5 years that it took between 2013 and November 2018 when the SDTM when version 3.3 was published. I will tell you, sometimes I tell people this because they say, oh my God, another version of the SDTMIG, what are we going to do? And I tell a lot of people, most of the differences that you think about when going between versions here, it's highly overrated. It's not that complicated when you upversion. And even if you're on a version and you want to submit your data to the FDA and tell them this is the version you're using in your define file and everything like that, but you can still take advantage of some of the new stuff that's in the SDTM implementation guide for the next version. Whether it's custom domains or non-standard variables instead of standard variables, you can implement this stuff. So what we're going to focus on today is the agenda I'm going to cover today is updates to the SDTM, because obviously the SDTM is the foundation for the implementation guides. Then I'll talk about changes to the SDTM implementation guide and talk about these general areas. And in the event that I get finished ahead of schedule, which almost never happens, I have extra slides at the end. John said, you sent me an awful lot of slides. I said, yeah, just in case I have extra time to cover all, I brought some supplementary slides. Version 3.3 was in the works, actually started back in 2014. So 2013, we had version 3.2. Not very much longer, we were ready to release batch 1 for public review. We decided that 3.3, rather than save everything up until the end, and so that this big package for public review is the whole SDTM implementation guide, we submitted it in 3 batches. And so batch 1 went out early in 2014. Batch 2 went out in November 2014, and wow, it looked like we were on a really aggressive schedule to get the next version of the implementation guide out really quickly. But guess what? We didn't. Okay, so it wasn't until 2016 that the third batch went out for public review. And then since 2016, in 2017, we had some additional comments from the FDA that caused us to try to resolve FDA comments. So it took a while between September 2016 and November 2018 before we got everything in order. There's another reason too why the timing is different. And then, so this was just the rationale for the batches. We decided that it would be easier to just do, people could review a third of the content during the review period instead of looking at all at once. For the people who aren't SDTM people, and even for the people who are, just as a reminder, that we have the model. It's the SDTM. It's a relatively short document, like 40 or 50 pages or so, depending on how you print it out. There are two major implementation guides. There's the SEND implementation guide for non-clinical data. That SEND is Standard for Exchange of Non-Clinical Data, that's SEND. And then the SDTM implementation guide for human clinical trials, which most of you from clinical understand pretty well, at least in some version. Then we have these implementation, what I call supplements. Now, not everybody calls them supplements, but I do. And I think no matter what you call them, there is this hierarchy. That none of these documents here, this is SEND-IG for animal rule studies, which just finished public review, or is finishing public review, I think in the next week or so. The SEND implementation guide for developmental and reproductive toxicology studies. The SDTMIG for medical devices, the one for pharmacogenomics, and the one for associated persons. None of these at the bottom are standalone documents. Because I call them supplements because you still need to go back to the parent implementation guides in order to get all the principles that are evident for a submission using one of these implementation guides. Versioning. What was then the SRC and the governing group at CDISC decided that every time an implementation guide goes out, there's going to be a new version of a new version of the model that goes out with it, because just about every implementation guide has new variables, make up new tables, new concepts. So in order to make sure that the model is up to date with the implementation guide, we have the implementation guides. Every implementation guide is tied to a version of the SDTM. At the same time that 3.2 went out, the SDTM implementation guide for medical devices version 1 went out, so they were both based upon the SDTM version 1.4. 1.5 is explicitly for the SEND implementation guide version 3.1, and so forth. So this is how they're aligned. A little bit of the history in terms of when did all this stuff happen. We started way back when with version 3.1, back in 2004. That was the first version that the FDA had included in the study-based specification within the ACTD. And Pfizer had made the first SDTM-based submission back in 2004 using version 3.1. That only had 23 domains. And you can see that each version of the implementation guide with 3.2 had 46 domains. And now we have here in 2018 and 2018, 3.3. There's actually 57 domains in there. I counted it like 10 times. And each time I got a different answer, but I think I definitively counted the last time it was 57, but I didn't get to update the slide. A couple of these did have domains because they were actually, as I mentioned before, developed for other implementation guides that weren't the SDTMIG. So what do we have going on with the SDTM? In version 1.5, we have new tables here, and I bolded the ones that I'm going to specifically talk about. The subject disease milestones and the trial disease milestones, I have a bunch of slides on disease milestones because I think that's an important concept in 3.3 that I really want to spend some time on. We have domain-specific variables, and I've got a slide on that. And then I'm going to cover a little bit of this stuff here. This implementation, this SDTM version was solely for reproductive toxicology studies, so probably not of interest to most of you that are here today. If you are, we can certainly talk about it later. The concept of domain-specific variables initially involved with this medical history event date type. For example, some people didn't know how to represent multiple types of dates for a medical history condition that was probably the disease of interest for the study. The diagnosis date, last date of relapse, symptom onset date, those kinds of things allow a submitter to create multiple medical history records with the same MH term. That was the solution. That's the only place we really saw an implementation of this event date type. Method of administration came along. This EG beat number came along for when the tau was developed for actually conducting QT prolongation studies. And then this again was a DART one, and then these were added to update when we had minimal inhibitory concentration in the microbiology susceptibility domain to add those. But those are the only places that the team, the SRC, and now the World Governance Group foresaw those variables could be used, so they're domain specific. You can see we added a whole bunch of variables to SDTM version 1.5. Some of these came in through the asthma tau, and a couple of those taus, that therapeutic area user guides that some of you might have seen. So they're called taus, the TAUG. And obviously some of this came in from the repro, the DART guide %. And so did the nominal day and nominal label. Some of these, not all these variables that are in 1.5 are suitable for use in human clinical trials. And there's notes to that effect. I've actually got a slide to that effect, time to present it. But it's also an implementation guide. There's a table that says, hey, these variables aren't to be used in human clinical trials, even though they're in the SDTM. One of the things that was really important is focus of study-specific interest here. This variable has no domain prefix. This variable was created in case there's multiple sites in a body where something might be administered. The concept initially came up with non-clinical studies, where sometimes on animals that there might be several different treatment areas, and sometimes if a dermatological thing, you have different spots on an animal's back, where you can apply different treatments to different locations. And you want to be able to have those treatments in EX linked to those treatments when you start seeing observations. And so this spoke ID variable allows for a focus area within a particular subject to be identified and tracked throughout the whole study. That's why there's no domain prefix for this variable. Its first implementation on the clinical side was in the ophthalmological examination data set. In 1.6, again, mostly reproductive toxicology studies for non-clinical. There was this one variable that came up, this non-host organism identifier. There was a variable that was being used in some of the pharmacogenomics domains called N species. That was dropped in favor of having this non-host organism identifier. It's used in the microbiology domains, but it's also used in some of the pharmacogenomics domains in the SDTMIG PDX. It allows one to identify an organism and then provide an additional table the taxonomical classification of that organism. I know that sounds really complicated, but it's not that bad. We have in 1.7 these additional variables that are companion to the SDTM version 3.3. And this is some of the new stuff that got applied into the SDTM implementation guide that we're talking about today. So, we talked about the SDTM. What's happening in the version 3.3? I could have put in probably like 24-point font, or maybe this is actually 20. I probably put it at 72-point font. The publication format, it's no longer a PDF. It's HTML. And while the CDISC website says that you can download the HTML file, and you can, the problem is that when I downloaded the HTML file, you lose the scrolling table. So if I access the implementation guide online through the CDISC website, there's tables that scroll. So remember in the PDF format, some of the tables had like 3 different blocks because they were so wide. You just chunk them up into either a landscape or a portrait page. Required some making the tables kind of like wrap around. That doesn't happen now because there's a scroll bar, if you go to the online version, that allows you to scroll left and right, which is not necessarily terribly convenient, but it works. The problem is when you download it into HTML or try to print it into PDF or do something else with it, you lose that ability to scroll, and so the variables that are way over here on the right-hand side kind of get chopped up. And if anybody on the phone or in this audience here has successfully figured out how to download the HTML file and keep the scrolling in, I greatly appreciate it. One of the big things that changed here was the approach to morphology and physiology domains. Back in 3.2, there was one morphology domain. The thinking at the time was all morphological things can be represented in a single domain. This was following the precedent set by the SEN team, where the SEN on the non-clinical side, they had organ measurements and they had some of the macroscopic examinations observed in necropsy. If you combine them together, they would have been the SDTM implementation guide morphology domain. The rationale was, okay, SEN can do it with two domains that aren't body system domains. Can we, in the clinical world, do it with one domain that's not body system based? And then the thinking was also that there would be body system based physiology domains. So it was cardiac domain or a respiratory domain. The problem was that it had to be really difficult to separate out what was physiology and what was really morphology. And sometimes morphology and physiology were very closely related. So the thinking reversed itself. And then we developed for 3.3 body system-based morphology and physiology domains. So all the morphology and physiology of a single organ system goes in one place. There were a bunch of minor format changes here that are listed. They're also listed in the implementation guide, changes from the last version. Not too particularly important in my mind that you can't research on your own. So this is what I just talked about. The single domain morphology, 3.3 actually, so 7 so far, and I know more are coming because there's more therapeutic area user guides coming out for different diseases that are going to be targeting different organ systems. And lastly, even though the morphology domain is in 3.3, there is a note there that says it's going to be deprecated. So anybody that's implementing in the future should be trying to implement the body system-based domains, and don't go any further with MO. But if companies implemented MO and it's still working, then they can still use it. So these are the new domains that appear in version 3.3. In addition to the body system-based domains that we had here, there was also functional tests. And functional tests is designed to be set up much like the questionnaire domains, with the category being the name of the test. We also have these domains for subject milestones and trial milestones, which I'll be talking about, as well as these intervention domains, one for meal data and one for procedure agents. Okay, so what happened between 3.2 and 3.3 was that this RS domain was expanded to include data that would have been in clinical classifications. The clinical classification domain was another questionnaire domain. It's like Child-Pugh scores or PASI scores or things like that really aren't questionnaires because they're assessments. And the QRS questionnaire team decided that they were going to have 3 different kinds of tests. There were functional tests, which again weren't questionnaires. 25-foot walk test is not a questionnaire, but it had been under QS. Okay? The clinical classifications really weren't questionnaires because there weren't any questions. They were just assessments. And so that was also another subdivision of what had been data that would have been represented in QS. And then and then there was the original questionnaires, the stuff that is questions and has answers. So what the team had was some of a challenge trying to assess, well, which is part of disease response and which is clinical classifications. And there are bumpers and splitters on the SES team. There are people that say, we should have kept 2 domains. I would have done that, okay? But there are other people on the SES team that say, hey, this is kind of like the same kind of stuff. They kind of look the same. Let's lump them together. The only problem we run into there, of course, is that now we have to use the category variable to describe something that would have been a domain definition level description before. The tumor domains were also expanding. rather than being limited only to oncology and tumors of interest, now they're also expanded to include other types of lesions that are not cancer. This is the QRS thing that I was just talking about here. So in 3.2 we have QRS, and now we've got these 3 other domains that are now children of that. One of which is the original questionnaires, and then the other 2 are the responses that includes clinical classifications and the functional tests. In the implementation guide version 3.2, there's advice that says that if you have a subject that was a screen failure, or they were randomized, or they were not a screen failure, but they were never assigned to treatment, to populate the arm code with values for screen failure and for not assigned. The FDA in the technical conformance guide, however, said, hey, include records and demographics for these people, but leave the arm codes blank. So what we did is provided a mechanism for, in these 2 variables here, variables are added to demographics that allow a user, an implementer or submitter, to describe why the arm variable is null. For example, screen failure. They were never randomized, or not assigned. They made it into the trial. There's additional advice in there for subjects that maybe only completed one stage of a study and didn't complete the next one, in terms of how to represent the arm code. I'm not going to go into that because that's not different from what it was in 3.2. Okay. All the other people in the room? No, not too many? Okay. Well, if you weren't at a person in this room, now Sandra's there. When she sees study population flags should not be included in the SDTM data, we all clap, right? Okay, because for the longest time, we've been trying to get the FDA to agree, not put this stuff in SDTM. It belongs in ADAM. Okay, these population flags for intent to treat or completers or per protocol population, they don't belong in the SDTM because they're actually done later. And what companies are actually doing was taking these data from the ADAM data sets and putting them back into demographics. And that creates a huge traceability problem because everything in ADAM is supposed to be traceable back to the SDTM. And something kind of breaks when something in ADAM traceable back to the SDTM is actually traceable back to ADAM. It's got this circle going here, so it doesn't work out too well. It took us a long time to convince the FDA that this circle didn't work out too well, and they finally acceded to our demands. And now there is a statement in there that says keep them out of DM and a description of how they can represent in the data sets. That description was actually there before, but now it's much more important to understand that that's where they get represented. And so accordingly, these standard supplemental qualifiers that were part of this variable in demographics, with the subcolumn variable in demographics, and the corresponding example in section 8 were removed. So we're done with that. Isn't that so nice? The changes to baseline flag. Baseline flag was a variable that's been around for a long time in the SDTM implementation guide, the SDTM. It was sponsor defined. That's a problem when every sponsor is defining their baseline differently. And again, this was something that we pushed the FDA for a long time. A lot of companies were taking baseline flags determined in their ADAM data sets and bringing them back into the SDTM data sets too, violating that traceability aspect again. So what was done was some companies that were actually populating the VLFL variable as sponsor defined aren't prevented from doing it. We didn't want to like chop everybody off and say you can't do that. But it was changed permissibly, okay? And that was a problem. So what we added was this new version called lapse observation before exposure flag, okay? And LOBXFL, say that 10 times fast. That's what it is. And it was added, we wanted a consistent definition, so it's not sponsor defined. It's defined constantly for everybody across the industry. It is the last non-missing value prior to the RFX STDC for the subject and demographics. So, not only does it have a constant definition, it's also being generated based upon data that's in the SDTM, not coming from someplace downstream, upstream, downstream. Which way is that, upstream or downstream? I always get it mixed up. Anyway. This is not too spectacular. A new section was added for how to populate epic variable, because you know in the technical performance guide, the FDA is asking for companies to populate to add an epic variable to a lot of the data sets whenever possible. And so even though the epic variable, or epoch variable as some people pronounce it, is not in all the domain models, we did provide guidance on how to implement it. Was there a question back here? Yes.
Sure. So that means all the baseline flags should be dropped from the CDISC starting with that new version.
So the question was whether a company should start dropping baseline flags. Here, okay, that's more implementation advice that I would give as a consultant. I think that all I can do here, in the absence of knowing more importantly about what companies' processes are and how they're created to begin with, I can just say, this is the recommendation. You need to figure out how to manage it yourself. Okay, and because it's going to be different for every company. The companies that were doing it right are probably going to continue to do it right using BLFL, and the companies that were recycling Atom data back into the SDTM for test purposes might think about doing it differently. That's all I can say about it right now without knowing more about the process.
Sort of related, what about keeping emergent flags?
But keeping?
And their drive,
Oh.
They keep asking for them in SDTM.
We didn't actually, there was no changes that I'm aware of to the treatment emergent flag. It's still a supplemental qualifier for adverse events.
Okay.
I don't think I didn't see any change to that in 3.3 versus 3.2. I know it's still a subject of ongoing discussions with the FDA. Maybe it'll be in 3.4. It is a supplemental qualifier, so unless the reviewer is particularly looking for it in the SDTM data, they're going to have to go to the data sets to figure it out.
What is the feedback from FDA regarding dropping or making baseline flag permissible and also
Oh, so the question is, what was the feedback from the FDA on this? And the feedback from the FDA was great. Okay? We tried to do this, I think, prior to version 3.2, right, Sandra? So we're very happy at the fact that the FDA finally agreed to let the baseline flags be derived from the SDTM data and not from Atom data. It took a long time to get them there, but they're happy with this now. And the other thing too is that the reviewers at the FDA are becoming much more proficient at using the Atom data sets for their intended purpose. And I think that, you know, Sandra, her experience in the Atom team realizes that the FDA is taking much more active involvement and understanding the analysis data sets much more so than they did before. And there's even a workshop on increasing the efficiency of analysis data sets coming out in June. Yeah. So it's a public meeting. You go to the FDA website, you can find out that they're having a public meeting on increasing the efficiency and usability of analysis data sets. And so they're becoming much more comfortable with using them. And so when stuff is missing from the SDTM data sets, they say, oh, I know where to find that. This was a clarification. Maybe over here. There was a clarification in the values that are used for the relative timing variables. So these are the variables, the STRAF, NRAF, the STRTPT, and the NRTPT. In 3.2, there was some type of a typo. And nobody can figure out how it got in there. It's like, who did that? It's like, no, they did it. No, nobody did it. But somehow it happened. These values, coincident and ongoing, were inadvertently added to the allowable values in the implementation guide, because it's all the same codes. The implementation guide says that, you know, for these variables, use these values, and these other variables use these values. Well, the mistake was made here. So in version 3.3, it reverts back to what it was in 3.12 and 3.13, to have the values of coincident and ongoing removed as recommended values, even though they're in the code list. A new section was added. I'm not sure how would you want to. Anybody start the watch, John?
You've got like 10 more minutes.
Okay. So this new section was added, section 9. So for those of you who are familiar with the old 8 section SDTM implementation guide, with the last section being section 8 for the relationships of the qualifiers and right now, there's another section that's called study references. And the study references right now, it's, I won't say a little bit arbitrary, but the rationale for why each one of these in there is not, what should I say? I think we have device identifiers and non-host organism identifiers as tables that were previously in, this was previously in the medical devices implementation guide, but the device identifiers data set is being used more and more in other non-device based human clinical trials to put in device settings and device information. So the SDTMIG for medical devices was originally created for the submission of medical device data on approval of medical devices, the 501Ks and PMAs for medical devices. However, right now there's nobody at the FDA that regulates medical devices that is requiring anybody to submit an SDTM-based format. However, a lot of the therapeutic area user guys have been including device settings when certain measurements were made. And so they've been borrowing from the medical devices implementation guide and putting those into their normal biological and drug submissions to the FDA. So this is brought up to a higher level. It actually appears in the SDTM implementation guide now, so that people can see how that's actually implemented. The non-host organism identifiers, I've got a table and I'll show that for that in a second. And the other one I'm not going to go into here was just these pharmacogenetic biomarkers. So this is not subject-based data, but it's database data. There's databases for genetic anomalies and for when different nucleotides are altered, SNPs and other kinds of the deletions and other kinds of things that happen. When there's combinations of these things, they're known about their database. So someone says, well, you know, this protein's missing this and that protein's missing this, or this protein's missing these amino acids in these places because of these changes in the DNA. Then we've got, sorry, the shadow. This documents the known about findings in the database that apply to the subject in a particular trial. There's also an SP domain in the SDTMIG PGX. I think I have a slide later on the non-host identifiers, but it's not going to apply, I think, to most people that aren't doing studies that involve testing for microbial susceptibility or other types of diseases that involve characterizing microorganisms. Disease milestones. All right. This is a new concept. It first came out with the therapeutic area user guide for diabetes. And the first characterization we had of this was when subjects were having hypoglycemic event. When a subject has a hypoglycemic event, a bunch of stuff gets triggered. This is also going to apply to lung cancer, and I think also to diabetic kidney disease. So there's a couple of other trials that are actually using this implementation. Trial milestones. It's a trial design table. It's in section 7 of the implementation guide. It joins the other trial level data sets because you know what these things are before the trial begins. They're planned. You plan to collect data. The subject milestones data set is like subject visits. This is how it actually played out for each subject. So what happened is we added the trial milestones data set, which, like all the other trial design data sets, appears both in the SDTM and the SDTMIG. Subject milestones, likewise, in both places. And then new timing variables that I had mentioned previously for the general observation classes. So let's take a look at an example. In this particular case, this was the one from diabetes, from the top diabetes. When the subject had a hypoglycemic event, that was going to trigger a bunch of data. So it's declared in the trial milestones data set, which is pretty small. And if it's a pretty simple study, this is all it is. It's the study ID, the domain, what it is, and then what's the definition of that. How are you going to determine when a trial milestone has been reached for a particular subject? And this variable here says it's repeating, which means that a subject can have more than one of these milestones. That's all it does. It's just a flag. And this is just a partial record. We can see that this particular subject actually had 2 hypoglycemic events. events. And we have the timing of when they started and when they ended. This is the characterization, but most of this information from this data set, much like trial visit, much like subject visits, comes from the data that you collected. You don't get it until after your study is over. And so we have these additional variables added to the general observation classes. This disease milestone instant name. And you notice that was hypo 1 and hypo 2 because the subject could have more than one. We have the RELMIDS. This is the temporal relationship to the milestone instance, whether something happened before or after, whatever that milestone was. It could be retrospective, coincident, or after that. And then sort of the DTC for that disease milestone instant date time. Let's take a look and get an example. Here's the hypoglycemic event we had in diabetes. Right? It happens. Bang. Okay. Start asking the subject, what might have caused this? Did they miss a meal? Did they do something different? Did they exercise more than normal? Whatever happened, right? There's a whole bunch of precipitating factors that are asked about. Like, let's ask about the most recent meal. Okay? What was it? What did they eat? Okay. When did you last take your insulin? Or other study method you're taking? What was your blood glucose level when this happened? When you first noticed this was happening, what was your blood glucose level? Okay. And then what medications were taken to treat it? In many respects, this is just like a visit. Okay, subject shows up for a visit. We have a bunch of data we're going to collect in the CRF when the subject shows up for a visit. This is just like a visit, except we just don't know when it's going to happen. It triggers a collection of a whole bunch of information. So how do we represent that information? This slide shows a little bit more about all these things we just talked about that are happening, but I kind of blow it up a little bit, so maybe people in the back of the room can get a little bit of a glimpse of this. It's hard to show everything. But I want to show that we have in these different domains here that are color shaded, we have this MIDS of variable, okay? Hypo 1. We know that everything that's labeled with hypo 1 is related to this first hypoglycemic event that the subject had. Because remember, this could be repeating, there could be a second one. We have this MIDS-REL variable. And actually, I think it's REL-MIDS now. There was earlier versions that had it the other way around. But, so this is a snippet of data from an older presentation, an older model that we initially did at the time that the diabetes tower was being developed. So now it's REL-MIDS. This is correct, but this isn't. But the general idea is that anything that's happening in these blue areas here is relative to the event, to the milestone itself. And then we have the MIDS. These actually show up nice and yellow on my computer, but then the hypo ones don't show up very yellow here. But this data allows us to group all this data from multiple domains, group it all together with this event, just like we can with the visit number in traditional SDTM implementation guide domains.
So let me ask you, it's REL-MIDS or MIDS-REL?
It's REL-MIDS. REL-MIDS is the correct
So
one.
that one is.
Yeah, you can go back to the implementation guide, and I know I had it right on this particular slide here. Okay. It was just, we had changed the name in midstream while we were developing the diabetes tau, and this information here came from a paper I wrote at the time about this. And I think what did is I went into this paper, I actually went into the paper and did an edit replace, and for some reason it didn't replace that one. Good question. So this has also been applied to disease milestones for lung cancer. So lung cancer tau, which is going to be published very soon because it just finished the resolution of public review comments.
I just approved it yesterday.
Okay, so it's coming out soon, and I think I just approved it yesterday too, so it should be coming out any day now. But you can see that they had, in this particular thing, they're looking for things. Remember, we said that this doesn't have to be stuff that's only in the trial. It can be events that occurred prior to the trial beginning, prior to the subject's entry. So this particular case, they're interested in these milestones. So when they were initially diagnosed, when the metastasis occurred, and then the diagnosis of first and second progression. That means that for every subject, they're going to be collecting this information. So we have these subject milestones, And obviously, there's probably going to be TU and TR and RS data collected in conjunction with each of these, as well as maybe some of the general health domains and safety domains as well.
So are those two domains apply to all the disease or just those?
Repeat the question.
So I asked you to repeat the question because I didn't quite get it either.
Are those two domains apply to all the disease area or just those two?
So the question was, if these domains apply to all the disease areas or just this two? And the answer to the question is, these can be used anytime you're interested in collecting a mass of data when a particular event is triggered by a particular condition. So they're not limited. I was thinking more about hypoglycemic events of diabetes and diabetic kidney disease, also using disease milestones. I wasn't thinking, when we initially developed this, we weren't initially thinking about its application to cancer studies. But it turns out that this is a very appropriate use of this. And it illustrates the use of collection of data that occurred prior to the subjects being enrolled in the trial.
I just have another question. So this brings us to another point where the data that you collected for disease milestone is from existing panels. And then you were required to interpret that data to trigger all of this information to pull into the disease milestone. So does that require a lot more derivation in SDTM?
So the question was, does it require a lot more derivation in the SDTM? And the answer to that question is, in my view, personal view, is no. Because it's just like a visit. The subject shows up, you do the labs, you do an ECG, you ask them about adverse events that happened since the last time they were. You have a bunch of stuff that gets triggered. This is the same thing. Now, will you have additional data in the lab domain that doesn't have mids and mids-rel mids on it? Yes. It'll be mixed in with all the other lab data, but you're able to filter out the lab data that was collected during a hypoglycemic event versus the lab data that was collected during a normal study visit based upon the timing variables.
But a lot of times when you collect the data, the evaluator, the investigator will not be able to combine all of this information together to figure out whether it's triggered or not. It has to be done in the sponsor site.
Well, the thing is that it's important. So the question was about the investigator. This is clearly defined the definition of the trial milestone. So it has to be clear to all the investigators in the study at all the sites when a hypoglycemic event is happening. And we can get into the splitting the hairs about how do they know that stuff. But the bottom line is, I'm thinking that if this is specifically defined, when the blood glucose concentration falls below the specified level, whatever it is, okay, that's pretty objective. It's a pretty objective criteria. Someone knows, hey, it's time. Now it's time.
Almost. We've got some questions from the.
Oh, okay. So let me just wrap up here real quick. Okay, that was just a new section that was added. In case anybody looked at the domain specifications and didn't know what all the columns were, there's now a section that tells you what all the columns are. This section, this table, which was one of my favorites in section 2.5, is taken out. These are the variables I talked about. This is actually represented in table format in the implementation guide, what not to use and what to use with caution. And these additional text on 200 character variables is all combined in one place in a table. This is not the table, but this is a synopsis of the table. It's in the implementation guide. And additional text was added around permissible variables because people are still very confused about them. And bottom line is that, you know, if you're collecting data about something and it's a permissible variable, but there just happens to be no data, you should be including it in the data set, okay? Because it's on the CRF. And if you take it on the CRF, tell us in the study data review guide that, hey, this question wasn't answered by anybody in the trial, therefore it's all the data is blank. But it's still there because it's annotated on the ACRF. Oh, I got to the point where I have additional slides as time permits, and guess what? It didn't. So there are some additional questions. So, Dave, what's going on?
Okay, yeah, so we've got a few questions from the webinar that I wanted to cover. Number one is, are the disease milestone data sets only used for milestones related to the disease? And is this the only difference from DS, I suppose, the protocol milestones?
Yes, so these milestones are very different than the protocol milestones, which are generally going to be followed by everybody as part of the trial design. These disease milestones are going to be generally, all of the use cases so far have been involved in disease understanding. And they're particularly around characteristics of the disease understudy. That's why they call it disease milestones and not protocol milestones, because they're only the disease. And there may be subjects in the trial that never have triggered any event. The subjects want to be in the we're very constantly talking about never have a hypoglycemic event, but we know that everybody's going to have a protocol milestone of a certain type. It's going to happen.
Okay, great, thank you. And one more. When using the SM dataset, should we double record initial diagnosis there and in MH, or should it be recorded in one place only?
Tough question. I won't take the fifth, but when I have insufficient data, I'm hesitant to provide a response. So I wish I could.
Okay, fair enough. I guess that one's for a later date then.
Yeah. Was that it?
Yeah, that's all I got.
Okay. Maybe just about time?
Just about time.
Okay. I'm going to give this back to you, and you can do whatever you want to with it, with Ron.
All right. So our second presenter, and we may need to do a little bit more adjusting of sound here or something like that. Second presenter is Ron Ganapathy from Genentech Roche Genentech. I never know which one to say first. And you know, I got to say, I actually was an employee of Genentech. I was the 350th person that they ever hired. That was long ago in a galaxy far, far away. And one of the things that's way different now than then is that after Genentech was acquired by Roche, the two of them together have become the fourth largest pharmaceutical company in the world. Right down the street, in terms of revenue, and in terms of people employed, it's it's in the tens of thousands, yeah.
It's 100,000.
Yeah, 100,000. Okay. So one of the things that really impresses me about Ram is that he has undertaken this project, which to some of us just seems impossible, which is to impose order on a huge array. How many drugs? How many indications? It's a lot.
More than 100 studies.
More than 100
200.
indications?
I would say studies, not indications.
Okay, so you might want to look into that because historically, 10 or 12 years ago, the top 10 pharma companies in the world were running, depending on which of the top 10 it was, anywhere between 1,200 and 1,600 studies concurrently each year. And I got to believe that Roche isn't far off that mark. You may not see all of them, but it's a lot. It's a lot. It's a lot of data, and it's a lot of stuff coming from a lot of directions. Anyway, Ram actually has some pretty impressive credentials for addressing this, because he's got a background in CRO as well as pharma services, and also analysis as well as data management and SDTM production. So I think I've talked enough, and with no further ado, I will begin Ram and switch over to his slides.
Okay, thank you, John. And thank you, Fred, for a very informative session on SDTM 3.3. It's really impressive, and it seems like we have a lot to do when we are portioned from 3.2 to 3.3 across all companies. So today I am going to talk about, Fred talked about the standards, and now I am going to talk about the implementation side of things. How standards are implemented in big pharma companies like Roche and Genentech. So my topic is on putting SDTM at the heart of Roche Genentech drive towards fair data, and I'll be talking about a project called EDISC. EDISC stands for Enhanced Data and Insight Sharing. So EDISC is an enterprise-wide initiative that is ongoing at Roche and Genentech to make our internal data fair. And CDISC SDTM model is at the heart of the work towards the prospective and the retrospective studies, and it is also spans across all our early stage and late stage clinical studies. So our agenda will talk about Roche and the pharma data governance at Roche, and I'll give a brief introduction about what FAIR is, and then we'll talk about the problem statement and prospective and retrospective view of things. So Roche is a global pioneer in personalized healthcare. So we have two divisions, diagnostics and pharmaceuticals, and we have like more than 90,000 employees worldwide across both diagnostics and pharmaceuticals. So it is very unique for a healthcare industry to have both under one place. So we have three R&D units. So one is the Genentech at the South San Francisco here, and the next one is at Roche in Basel, Switzerland, and then the third R&D unit is in Shugai in Tokyo, Japan. So, so Roche is one of the #1 R&D investor in healthcare, and we have 30 different R&D sites worldwide, and you know, like our diverse approach helps us to maximize innovations across different organizations and locations. So we have footprint almost in many countries, and even biometrics exist in like across all different time zones from China to Europe and different time zones in North America. So our culture is like highly decentralized model. So that means like Roche, Genentech, and Shugai operates with their own processes and tools and people and resources, and we have like diverse thinking. So we are encouraged to conduct experiments, learn from it, and develop innovative ideas towards supporting the drug development. And at the same time, we focus on agility, stability, and flexibility and speed and how we do things. So with that, I'll move on to the pharma data governance. So the pharma data governance sits within the biometrics of the Roche company, and this is the body that creates and governs the standards that are adopted across different functional units within Roche. So we have organized ourselves as verticals and horizontals. The verticals are the therapeutic areas, oncology, and I2O stands for immunology, infectious disease, and ophthalmology and neuroscience. And we have horizontals. The horizontals are the domain teams. So the across TA domain team consists of the data standards that widely used across all therapeutic areas. So it includes a suite of data standards that can be used for collection, tabulation, and analysis that can be used across all therapeutic areas. For example, like demographics, adverse events, and these fall under the across TA. And the specialist domain teams, they either concentrate on a specific indication or they concentrate on a specific data type, like genomic findings, histopathology, or like it can be like a specific indication like Alzheimer's. So all these domain teams are formed with cross-functional SMEs with representation from clinical science, biometrics, safety, and across all different organizations within Roche. And we create standards and we govern standards on how it is implemented across all studies. So the Roche data standards covers from collection, tabulation, analysis until submission, and it includes all layers of implementation, all the biomedical concepts from terminologies and the metadata and the tools and the processes that are required to implement the standards at a study level. And we are fully aligned with the CDISC data standards, the SDTM and ADAM, and like the SDTM and ADAM standards are applicable for all the studies for consistency across all submissions from Roche and Genentech. So in this slide, this slide talks about our metadata repository. So we call our metadata repository as GDSR, so Global Data Standards Repository. So it is our MDR for collection, analysis, and tabulation for biometrics, and the functions outside of biometrics are also use the GDSR, like the protocol development and the clinical development plan. So the GDSR can support different functions within Roche and Genentech, and the GDSR can interact with our external data sources like NCA and CDISC library and stuff. So the GDSR is built on an RDF data model, and it has REST APIs built on it where you can electronically get and transfer information outside of the metadata repository. So let's pay fair. So it is all about EDIS and what we have done in EDIS with respect to SDTM, CDISC SDTM standards. So I already have mentioned FAIR a couple of times, and it is gaining traction within the industry these days. So the term FAIR originally originated in 2004 in a workshop in Leiden, Netherlands, with representation from academia, industry, and other agencies. So FAIR is a set of guiding principles. By following those principles, you can make your data reusable across like multiple divisions within the company. So FAIR is like very easy to remember from my view. So F stands for the findability. So findability is like where is our data. So our data should be highly curated and annotated with rich descriptions so that it is easier for us to find the data on where it is, and it should be accessible. So with the right amount of access restrictions, so anybody when they want to access the data, they should be able to access the data electronically. And the interoperability piece ties in closely with the data standards. So interoperability is the use of standards and consistent terminologies that enables the data for easier integration and to create analyzable data sets very quickly. So if we follow F, A, and I principles, the data automatically becomes reusable for its primary use and many of the secondary uses. So okay, so why do we have to do EDIS? So what is the problem statement behind that? So I have come up with a simple example that stand by it. So a scientist has a question like, do you have whole gene sequencing data for patients with non-small cell lung cancer who had no history of smoking across all studies from Roche and Genentech? In today's world, you know, like we may need a month to get back to the scientist with that information, and you know, like it is not just one person effort for a month. So we need to put a team together, and they have to find the data, where is it, and they have to access the data, and they have to curate the data in a way that it can be analyzed to create insights for further scientific research. So you know, like this type of information should be available at our fingertips, and we should be able to you know, like readily get back to the scientist on these type of questions. So our CEO, Dr. Severin Schwan, states that, you know, like the way we currently operate leaves the data in a state where it requires months to access, pool together to answer some of the important research questions. And this is the valuable time in our patient's life. And you know, like we should do a better job in keeping the data accessible and curated and readily pullable to answer such important scientific questions. And Dr. Jeff Helterbrand, the global biometrics head at Roche Genentech, he states that, you know, like generating insights is not always guaranteed, but finding and accessing our data should not be a rate limiting factor in advancing science. So in today we have difficulties in time, and it is very difficult and time consuming to find, access, and integrate our medical data. And we have invested a lot of money and effort to generate this data. So these data assets include the clinical trial data, genomic, imaging, and other biomarker data, and our data assets like Foundation Medicine Flatiron, and the external data assets like TCGA, Genomic England, etc. So with that in mind, EDIS was created with the vision that the EDIS is actually a key enabler for the PHC 2.0 strategy at Roche. And EDIS is actually, it's not a system, it is a new way of working. So it is a new way of working to support reverse and forward translation. So reverse translation is what, like using what we have learned in clinical studies and real world clinical practice to further enhance the early research and development process. And forward translation is what, like we feed it back to the clinical trials and the clinical practice. So EDIS is a new way of working, and like we are looking to produce the data that fits tomorrow's that fits for purpose, and it also fits the tomorrow's use for the primary and the secondary use of the data. So we often think that the data life cycle ends at the CSR. So once the study closes, we think that the use of data ends there, but it does not actually end there. So there is a secondary use of data where the same data can be reused to answer several scientific questions. So EDIS is a key enabler to that. And EDIS is a 3-year program, and like hundreds and hundreds of people are working in this program across Roche and Genentech to support this. And a key strategy is to build the data capabilities and processes and technologies to support creating insights from data. So as you see here, the raw and the processed data or the foundation of EDIS. We call that as generating fair data and data management. And by utilizing the fair data and advanced analytics, we gain more knowledge and insights from the data that leads us to decisions and advancing scientific research. So the data layer at the bottom includes the diagnostic data, the clinical trial data, and the real world data. So I am happy to tell that like the CDISC SDTM is chosen as a preferred data model that is part of the foundation layer from the clinical trial data, that is all the retrospective studies and the prospective studies going forward. So the 3 key objectives of the program is the retrospective, prospective, and the cultural side of things. And the retrospective view is to get maximize the value from the legacy data. So the legacy data is more intended towards answering specific scientific question to support the use cases, and the prospective view is about the data verification of all the incoming data. So the data should be ingested or acquired in a way that it is fair, and it helps us to for easier pooling, and it serves the primary and secondary use beyond the life cycle of the clinical study. And the third and the important component of this is the data citizenship culture. So where everybody within Roche and Genentech are encouraged to adhere to the FAIR principles to generate the fair data in the clinical study and in the clinical setting that they are working on. So first, let's talk about the prospective side of things, and how we have applied the CDISC SDTM principles to support the data purification of all the ongoing and the future studies. So our goal is to have all the incoming data to be fair by 2020. So what fair means? So fair means the data should be in a really good format where we are in a stage to pool the data as it comes in, rather than going back and doing like fitting the data for pooling purposes. So today we are working on building capabilities and process in place, so that automatically when the data comes in, the data is fair. So we don't have to go back and chase our tail every time and retrospectively fix things. So this means that all the incoming data in future will support 2 things. So one is the primary use. The primary use is to answer the primary objectives and the exploratory objectives as defined in the protocol. And the secondary use is to answer any key scientific questions that may arise in the future. So I'll concentrate on the clinical trial data and the project that we were working on, and I am working on as a team for more than a year at Roche and Genentech, on how we have applied the data verification and towards ingesting, acquiring the data going forward. So to start with, earlier we discussed that Roche is a highly decentralized company. So we had the early phase studies and late phase studies, and the studies had their own collection standards and tabulation standards. So nothing was done in a consistent way across the company. And to add on top of things, we had multiple rave URLs and multiple multiple rave implementation that created the SaaS on-demand data in different format across Roche and Genentech. And there was no harmonized process to create SDTM. And since our collection standards were different, we were not able to have a consistent tabulation approach to tabulate the data. So our tools and processes were different across early phase and late phase. So we always think that, sometimes we think that the SDTM data is not good, but actually the problem lies in the collection side. So the collection has to be done in a harmonized way so that it can be tabulated and analyzed in a harmonized and a consistent fashion. So and then we found that we did not have a metadata repository for early phase standards, and we also saw that the external vendors, when they send data to Roche, they were sending data in different format to different parts of the company, and and we never had a tool to assess how close the studies are aligned to the standards, so that we can use a lot of tools and the processes and gain efficiencies down the line. So we had a lot of challenges in this project, and we had one question: so why can't we all use same standards and implement them in the same way across Roche and Genentech? And the same standards are developed and released across the company at the same time, so that the study teams can make use of it and implement at the study level at the same time. So this led to the launch of the Global Data Standards Implementation Team in 2018, and our proposal was to harmonize the collection and tabulation standards across early phase and late phase to start with, and to extend it to across different part of the Roche and Genentech in future. So we had a lot of challenges, and we started working on it, and we came up with a lot of innovative solutions. Some were simple but very effective, but innovative solutions to address all the challenges we faced. So our early phase data collection and tabulation standards were not in a metadata repository. So our first step was to include them in our metadata repository, and our metadata repository is now a single point of truth for early phase and late phase data collection, tabulation, and analysis standards across Roche and Genentech, across the entire organization. So because our collection standards were not consistent, we have different sets of template programs and macros to create SDTM datasets across early phase and late phase. So we reached out across the aisle. It was a collaborative effort across the early phase and the late phase team, and we have come up with a consistent, harmonized process for implementation of standards. So now we have consistent template programs and macros to create SDTM datasets across early phase and late phase. So initially we were not having a lot of programs, so most of the things were done at a study level. So every time a study team has to copy it from a different study, or they have to reprogram it from scratch. So as part of this harmonization effort, we have created more than 200 plus SaaS template programs that can work across our standard CRF across early phase and late phase to create standard SDTM datasets for our standards. So it was highly inefficient, and there were more study specific updates has to be made by the study teams. So we have made our tools more efficient, and so the tools are smart enough to identify a source and map it to target, and so that enables a greater reuse at a study level. So previously, we had no easy way to compare a study to the standard and see how close a study is aligned to the standard. Most often we see that the study team will adopt the standard and they'll change something at a study level that break all the tools and processes that we have built, so that the teams are not able to use the standard programs and macros that we have built at a study level. So now we have come up with tools to compare the study to the standards and highlight where the study is discrepant, and where standards are adopted and where standards are not adopted, and what tools can be reused at a study level. So it was a collaborative effort. A cross-functional global data standards implementation team was formed with representatives from clinical data science, the clinical data management, statistical programming, and the biostatistics to help us to build the process and everything. So So we made sure that we included everybody. It was an inclusive approach. So we created one harmonized process that can work for both the early phase and late phase, and we updated the programs and our programs and macros so that it can work across like all the companies. And so our philosophy is data points that are collected should be cleaned and tabulated the same way across early phase and late phase. And we have created reusable tools and template programs so that the study teams can reuse them with ease. So how reusable are our template programs? So initially we saw that the early phase and late phase had different data collection standards. So we resolved that by creating a harmonized data collection standard across early phase and late phase. So that means a CRF will look exactly the same across early phase and late phase. But we had another challenge. We had multiple Rave URLs that created SAS on-demand datasets in different formats. So that breaks our SAS programs and macros, because we need to refer the variable names when we program them to map the source to target. So we analyzed the different Rave URLs that we had, and we found the differences, and we have updated our tools and macros so that our tools will be able to identify the source URL that the study is built on, and use and work accordingly. So today our tools will work seamlessly across the early phase and late phase and different Rave URL implementations we have. So if a CRF is like 100% copy from the standard, it is like 100% reusable. Obviously, we don't have to make any updates. And most often times, so our standard CRFs will cover a lot of different things, like 100 different combinations. And the study may not need all the different scenarios when those are implemented at study level. So the study team will often remove a lot of data collection fields from the standards when they are implementing at the study level. So previously, what we had to do is like we had to go through our SAS programs line by line and comment it out, or remove those lines where did not have a source to map into target. So we have created new macros and tools. So where the SAS programs are smart enough to identify if the source exists, and if a source does not exist, it will skip the mapping. A simple example could be at a study drug administration forms. So we'll have kit numbers and lot numbers in our standards. At a study level, the study team may not be interested in mapping the kit, in collecting the kit numbers. So the kit numbers are mapped to the exposure domain, a CEX kit and stuff. So when we don't collect the source, so we have, our programs will see if there is a source. If there is no source, it will just skip the mapping for that line and it will go to the next line. So the study teams do not worry about all the stuff that are removed from the standards. They have to only think about what they have added on top of the standards at a study level. So for any new CRF or a data collection field that are added to a CRF or to a non-CRF data source, so only for those things the study teams have to update their programs and specs at a study level. And we have created an R-Shiny app to exactly tell the study teams where they have to go and make the update, which program they have to make the change to include a study level update. So we have built REST APIs in our metadata repository so that we can electronically fetch information from our MDR that can be used further downstream. And still we are working on making further improvements to our MDR. So we have created an R-Shiny app to leverage the metadata repository and the study metadata to compare the two and to provide a bunch of very useful information to the study teams. The first thing that the study teams will be able to identify are the study specific forms and fields. So what the study team has created on top of standards that does not exist in standards. So so these things are created by the study teams for specific protocol needs. For those things we do not have a collection or a tabulation or analysis standards. So for all those study specifics, the study teams have to interact with the data standards group on how to tabulate these things and how to analyze these things, and they have to interact with the biostatisticians and the clinical data management, and to effectively to collect and tabulate and analyze these study specific components. So the R-Shiny app will quickly help the study team to identify these things. So these are the usually the most challenging part at a study level, how to deal the study specific things. Now the study teams has an easier way to identify what those are to deal them in an efficient way. And then we'll be able to give them the SDTM mapping for all the standard forms and fields in one button click. So once they upload the data to the RShiny app, they can download the SDTM annotation for all the standard forms and fields, which directly goes into the SDTM spec that they create at a study level. And we provide the control terminology for all the standard NCA SDTM code list, and we compare the code list that are used at a study level, both for CRF and non-CRF data. We compare it to what we have in our standards, and the standards will have the code list that are created from the NCA database. So we compare the two, and we give the study team directly the code list that will be part of the e-submission, for all the standard code list. And we'll assess how close the study is aligned to the standards. So, and we create metrics based on how much standards are being used at a study level. And another good important thing that we are doing is so we are making sure the latest lab and the QRS metadata are used at a study level. So our lab and QRS metadata are huge, and the teams always have had challenges in using the latest lab codes and everything, because it goes by when the study starts, when the first patient is enrolled in the study. So the RShiny app will compare the metadata used in the study, and it will compare the lab and the QRS metadata we have in our MDR. And if the study team has not used the latest ones, it will highlight them so that they can go back and update it at study level. So the RShiny app, this app will be used by different functional groups within Roche and Genentech, and it will be used at a study build stage before the database goes live. So the study data managers will use it, clinical programmers will use it, and the SDTM programmers will use it, and the data standards managers can use it. So I'll give a high-level overview of an architecture of the app and what it can do. So we have the study metadata, so where we have a source of the data collection forms and fields and the non-CRF data collection modules, and we have the control terminology at a study level. And then we have our standards repository, so where we have the standard side of things, where we have the standards for data collection for CRF and the non-CRF sources and the control terminologies. So we compare the two through API calls, and the API calls return a JSON or XML file format, and we are using the JSON file format. So JSON file format is a tree-like structure. So when we send a request to our GDSR or the metadata repository, we get a JSON file back from it, and we pass the JSON file, and we create a two-dimensional data frame, and the data frame is used to compare what we have within the study. And there are multiple things that has to be compared. So we have unique identifiers, we have labels, we have pretext and data format, and we have user data, coded data, synonyms, and submission value, coded value, C code, L code. So we have so many different things to compare between the study to what we have in standards. So once we made all the comparison, so when we find a match between the study and the standard, so that means the study team has used a standard data collection CRF or a non-CRF source at a study level. And so once the match exists, so we can provide a bunch of things to the study team. So we can give the data tabulation for all the standard forms and fields at a study level, and we can give them ESUB ready control terminology that can be used at a study level. And the major thing is we can identify discrepancies. So our fundamental concept is, so when we are adopting a standard at a study level to make the study highly interoperable, adhering to the I fact of the FAIR principles, so we need to make sure that the standards are adopted 100%. So it does not make sense to to adopt a standard and tweak something at a study level. They break all the tools and processes that we have built for the study teams to use. So the app will identify the discrepancies, and it will tell the study teams know where they have adopted a standard, but they have deviated from it, and it is a good chance for the study team to go back and correct it before the database goes live. And the next obvious choice is like when there is no match in our metadata repository, it means that the data collection module is created at a study level. So there could be two reasons. So one is like we don't have the standards available for that. So it is a good assessment for our data standards group to see where we need to concentrate on future standards development, or the study team just did not adhere to the standards we already have. So someone will from the standards group will review that and will make sure that it is truly a study specific. And if we have standards available that the study team can use, we could advise the study teams to go back and use the standards that we already have. So on top of it, we give a metrics to the study team where we assess the study interoperability, like how much the study is aligned to standards. So have the study team adopt the standards to its entirety when they're implementing the standards at a study level. So this is a lot of work that we did, and we had an army of programmers working with us and to create like 200 plus SAS programs, macros, and R-Shiny apps that goes along with it, and that can be used at a study level. And we did all these things in like 12 to 15 months time frame.
I was going to ask, how big an army?
I would say like internally we had like more than like 15 people work for us, and externally I would say like the same number, 15 to 20 people working full time on this project.
Small army.
So are you planning to share this tool and data access with the industry or any?
Yeah, so the architecture can be shared. So you see how we have done things, but I don't know if the code can be shared outside of Roche and Genentech. Probably it's a good, but I have seen companies they put our code in a public GitHub where other companies can make use of it. That is something that we can check with our legal and see if that is something that is doable to support the industry and to make sure that like adhering to standards is more important, right? So it is a good way to do it, promote it.
So I'm assuming you guys get data from other sources too, like CTEP or other agencies where they run their studies for you, right?
Correct.
So, do you guys have plans to make that data usable, because that data is a complete mess when you receive that data, right? Yeah,
that's true. So the question is around, like for fully outsourced studies, when an external agency builds the database and creates the database and acquire the data, tabulate it, and they just send the tabulated data to us. So currently, like those type of studies are not in scope for us. But in an ideal world, all Roche Genentech studies, we would like them to adhere to the standards that we have developed, and eventually, like I hope in future those studies will under the scope of this project.
Do you have any plan for the ADAM?
Yeah, so right now we concentrated on the SDTM side of things, and for ADAM eventually we already have a lot of tools and programs for ADAM. To create standard outputs and to create ADAM datasets. We call that as stream macros for ADAM, and but that is not in the scope of what at least I was working on for the past 2 years.
This doesn't give you the flexibility to try other EDC systems, for example. You're pretty much committed then to RAVE.
Correct. So yeah, the question is like we are pretty much, so this setup does not help us extend beyond the RAVE EDC data collection module. So in today's world at Roche, like more than like I would say like 99% of the studies, early phase and late phase studies, it's done in RAVE, but we are conducting experiments with other EDC systems too. So the fundamental concept is the identifier. So if you see in the slide, the OID is identifier in RAVE. So every EDC system will have an identifier. As long as the identifier matches to what identifier we have defined in our standard metadata repository, the entire process can be reused, and the tools and the architecture can be reused. So if the identifier falls apart, even within RAVE, we'll not be able to use any of the standard tools and processes.
We
which MDR do you use?
So the question is like, which MDR do we use? So it is an in-house built MDR. So it is built on based on the semantic technology RDF file format. So where everything is a graph, and the MDR system is in existence, I would say, like for like past 6 to 7 years maybe.
Okay, I've got some more questions from the webinar here. A couple of them from your colleagues at Roche, actually. How do you envision using your FAIR system on non-clinical trial data like RWD or diagnostics data where you don't have SDTM standards?
Correct. So right now the tools are more inclined towards mapping source to target in SDTM, and that's where it is. But we have been getting requests on how to do this in the real world data, and the data that directly goes to our research, where where need not be in the format that the biometrics consumes. So that is something that we have to think about it and see how we can extend this model to support the real world data.
Okay. Next. How do you ensure that the data out of your system is validated or mapped and derived correctly?
So we do not the architecture and everything supports the metadata level. So it doesn't actually look into the data itself. So when we say our MDR, our metadata repository is a collection of it has the collection and tabulation and analysis standards. So it's all built and linked together within the metadata repository. So like when a study team is using a standard CRF, so we already have the link from collection to tabulation and tabulation to the next step. So the only thing that was missing was we were not able to get the data electronically out of the MDR system. So it was in a PDF file where where we had to compare two PDF files and find it. So now with the API and the REST API enhancements, now we are able to get the data from our MDR in a mission readable format, where we can compare that to the study level, and our MDR is a single point of truth. That means like we are comparing against a single point of truth. That means it cannot go wrong.
Okay. When do CDISC and FDA issued compliance specs come into play? I'm guessing.
So Pinnacle 21, yeah. So when the standards are implemented at a study level, so the study teams use the R-Shiny app and the SAS template programs and the macros that we have created, and they create the SDTMB datasets at a study level. After the SDTM datasets are created at a study level, then they use the Pinnacle 21 to do conformance checking.
Okay, so this is a multi-parter. Is the study level metadata also stored in your GDSR?
No, the study specific metadata is not stored in the GDSR. All the standards, metadata, and control terminology are stored in the GDSR.
Okay. And where do you store the clinical data?
So clinical data will be stored in our programming environment.
And do you have a CDR within your company?
I don't know what a CDR is.
All right, yeah.
Clinical data repository. So yeah. So we are building one. We are building a clinical data repository.
Okay, couple more here. Okay, last one. What is your rate of error comments from the study team? So I guess problems with mapping, maybe.
Yeah, so I would say it's less than 5%, because the way we operate is, so we just do not create the programs and give it to the study team. So we have a standard study and we enter test data for all possible scenarios that can occur at a study level. So if we have a code list, we try to enter at least more than one option in a code list, and we try to create different subjects that follow into different paths at a study level, and we test it with that test data before we make the programs and macros available for the study teams to use. And it is still brand new. We are yet to hear more from the study teams, but until now, at least it is less than like 5%, I would say. Yeah.
Actually, I'm quite curious about your data standard governance process. So for a company like Roche, it's quite challenging because you have any therapeutic area. Even within therapeutic areas, study may have different different variations. So how do you, it's great you have a tool to identify the difference between the standard and the study, and also, let's say, how do you justify the deviation from the standard, and also, if there's a justification, how long would that take for you to review and approve the study for the design stage?
Yeah, thank you. It's a great question. So it is about when the study teams create the standards at a study level, how do you manage that? How long does it take to review and approve those changes? So we have a internal process to help the study teams with the study specific things. So obviously, like almost every other protocol will need study specific data collection form or a field. So we have a process lined out. So the study teams will create the data collection process, and they'll submit it to the data standards and our DSG group through our internal system, and our DSG group will review the data collection and the rationale for the study team to collect it at to deviate from the standard and to add on top of things, and they review and they approve it. So usually the turnaround will be, I would say, like it's like probably 1 to 2 weeks for every request. So it's been done for both collection and data tabulation. Yep. So how are we doing on the time?
We're getting close.
Okay. So I have just 2 more.
We start asking questions. Actually finish your presentation.
Yeah. I just have 2 more slides, and these 2 are like it's about the retrospective side of things. So how we have approached our retrospective studies that were built 8 to 10 years before. So this was one of the most challenging part, and we had our dedicated team of data curators and integrators working on our retrospective studies. And from the data standards group, we support the data curators. They are essentially like statistical programmers. So we support the data curators and integrators to maximize the value from the legacy clinical data. So the legacy clinical data, it is targeted towards specific scientific use cases. That means to answer specific scientific questions. So the use cases are formed so that like we can reuse the data collected from the previous studies to explore different scientific questions with the goal to reveal new insights for research and development. So similar to what we had for prospective view, we had a lot of challenges on the retrospective side of things as well. So like the most time was spent on finding the data and the associated metadata. And when we say the curation of legacy data, so like the 8 to 10 years before, we did not have a governing model like SDTM to collect and tabulate the data. So most of the time, the analysis data sets were created from the source data itself. And and there were not many code lists and associated with it. And like even we did not have code list for race. So for a study that was conducted 10 years before, so we have studies where the race was entered in a user enterable text box. You can see all different types of races in the user defined text box. So at a curation level, so what we have to do is like for all the terminology driven fields, so we need to see all the unique values and try to map it to one of the NCA code list defined by that we have in our metadata repository. It's not just that, it is much beyond that. I'm just giving one example of what a curator will do when they are curating a legacy data. So and then we so once we found the data, so we tried to map the data to SDTMIG version 3.2. So it was actually challenging to map the data that was collected 8 to 10 years before to SDTM, because like simply the data did not fit into the SDTM models. So and also we were not able to create a scalable model or a tool so that okay, you create a tool, boom, you convert like 100 studies to SDTM 3.2. It was not possible for us to do it. So what we did was like we created some reusable R packages to map the source data sets to the target SDTM models. So and also so before pre-SDTM, luckily at Roche we had something called as generic data model. So for all the studies that had the data collected in generic data model format, so we were able to map it to SDTM 3.2 and create R packages that can go and like map the data from GDM to SDTM32 for those studies. And we also tried some machine learning tools and techniques to map the legacy data as an experiment. And also, like rather than doing all the domains in 3.2, we concentrate on the priority domains that are needed to support answering key scientific questions when the clinical data is put together with the whole gene sequencing data and other biomarker data. So that reduced the scope of what we had to do, and that helped us to concentrate on the right data to generate key insights. And to check the quality and conformance data, obviously Pinnacle 21 did not work because it incorporates all the FDA rules. So we created a subset of Pinnacle 21 checks that can be used only across the curated data. And and we created R packages to check the conformance of those curated data with the subset of Pinnacle 21 checks. So yeah, so it was very challenging from a legacy standpoint as well. And our story continues. We are still working on it. It's a 3-year program. It is still ongoing. And going back to our initial question, so when the same scientist comes back to us in the future and asks us the same question, so do you have the whole gene sequencing data for all the non-small cell lung cancer subjects with no history of smoking across all clinical studies in Roche Genentech? We should be able to get back to them in a day. With all the with all the tools and processes and the systems that we are building today, it will help us to answer key scientific questions in the future. This is only possible by adhering to CDISC SDTM standards, and all the incoming data are collected and tabulated in a harmonized fashion. So EDIS is a foundational to transform. It's a foundational for PHC 2.0 effort across Roche, and so it is a way how we get there from Route 66 to Autobahn. It's a better and more efficient way. Thank you so much.
I think we're going to need to defer questions to the end, because we're going to have another shot at Ram and also DJ from Gilead Sciences. They're going to be, they're going to have a sort of conversation about this sort of stuff and the way it happens at multiple companies at the end of this. We're going to take a break now for for like 15 minutes or so. Let's come back at the top of the hour. But I wanted to just sort of give you a little bit of a sort of a preview of what's coming up. As we were planning this meeting, we came up with so many timely subjects, we couldn't get them all in here, not even close. And so we're planning another meeting in late September, early October, and we're going to try and get it. We're going to try and make it a conversation with the FDA. So we'll have FDA people, or at least one person, on the webinar, and interacting with people with various perspectives in the room. So we'll have biometrics people, obviously, but we'll also have regulatory publishing as well. And some of the topics that we want to do in that next meeting are going to be about the technical rejection criteria, which were finalized in the beginning of 2017, and we're just going to sort of speak for 6 or 7 minutes in the second half of this meeting, but we're going to devote some serious attention to it in the next one, because I'm sort of convinced that hardly anyone in the room knows what's coming down the road. The the FDA installed tripwires in all of the hallways that you're used to using and never paying attention to, and if those were being enforced today, which they could be on 30 days notice, then I'm going to guess that 2/3 or more of the submissions that everybody's sending in would fail, would be rejected, handed back to them at the gateway. So anyway, we're going to have a conversation about that. Who's going to the CSS conference in June? There's going to be a few people. So I'm thinking that we should, sort of all get together and get a conversation going with FDA people there at the Fuse conference, and also recruit some people to be, participants in the next meeting. The other thing that people need to start worrying about, like right now, is the use of WHO drug B3 format, which is, I think, a requirement for new studies like now. And if not now, then in a couple of months. And there's some very interesting problems that we're gonna see once we start implementing B3 format broadly. So anyway, there's some really cool stuff coming up end of September, early October. And so I will let you go because people are like going, oh, okay, I got to get out of here. And let's get back together at 3 o'clock. We're going to start. So our next speaker is going to be Sandra Minjoe, and many of you know Sandra because she's worked with a whole bunch of you, and I've worked with Sandra too at I don't know how many places. But one of the things that was extraordinary about how things came together for this meeting was that we managed to get the architects of virtually all of the standards that we use on a day-to-day basis in the same room, well, almost in the same room, because Sally Cassells is still coming up, and she's going to be on the webinar. But it's extraordinary that these three people have really been the driving, forces behind most of the standards we use. And so now we're going to hear about the ADAM standard, and there are some very cool things coming out of the ADAM team right now. And by right now, I mean like today and tomorrow and next week.
We'll hear more about that in the later part today.
And so anyway, without further ado, I think I will hand you over to Sandra Minjoe.
Thanks much. Yeah, so with Fred, who was here before me. Hi, Fred. The former lead of all that SDTM stuff that he just talked about, and I'm the current ADAM team lead. And then we've got Sally Cassells coming up, who Define-XML guru. Okay, so this talk I'm giving here is about integration. Later on today, when we get into the panel, I will talk a little bit about the bigger picture of what's happening with ADAM and all the other things we have going on. But this is a slide deck that was presented last week. Were anybody in the room at the CDISC webinar that talked about ADAM-IG version 1.2 and this integration piece? I see one hand, or two hands. All right. So this is the exact same slide deck, all the way down to the name presented by Deb Bauer that Deb presented last week. So she gave me the slides, and I'm going to walk through them today. She's the sub-team lead for the integration team. I am on the integration team, and I'm also the ADAM team lead. So I know a little bit about this stuff, not as much as Deb, but I think I'm going to cover it okay. If you want to hear Deb's presentation, to hear what she had to say with these same slides, that presentation is available on the CDISC website with her speaking to it. So I'm just going to kind of cover it. I'm going to go through it a little quicker today because I know we're kind of short on time. And I won't get into it nearly as much detail, but you have that if you want to go look at what Deb presented. Okay, so the agenda, what I'm going to cover today, it's talking about these integration structures, but we're going to talk about simple versus complex integration first, because with simple integration, we can make use of some existing stuff. But with the more complex integration, we really need some different tools to help us. So we kind of broke it up into two pieces there. And with each of the simple and the complex, we're going to give an example of what that might look like. And then at the end, we'll do some facts and kind of wrap things up. So when we started the integration sub-team of ADAM, we had a few rules that we kind of put in place before we started developing things, because everybody had different ideas of how to integrate. So the first rule that we wanted to make sure we followed was whenever possible, we used what was already existing out there in the Atom world. We weren't going to reinvent the wheel. We had things that were working well in a lot of situations. Could we make them work for us with integration? So that was our first rule. Our second rule was we wanted to stay away from data flow because so many people integrate in different ways. You hear people integrating at the SDTM level, you hear people integrating at the Atom level, you hear people taking Atom data sets and merging them together or starting with SDTM and creating an integration from that. There's lots of different ways that you can get to the integration. And this is not going to talk about all those ways to get there. What this is talking about is the structure of where you end up. How you get there is up to you. And it's that way through the rest of Atom. How you get there is up to you. We're giving you a structure that you are aiming toward. Okay? So we're trying to harmonize again with the Atom, trying to achieve that harmonization across the existing Atom documents and these new integrated documents we're talking about, and making use of names, labels, definitions, trying to make all that stuff consistent. So that was how we started. Okay, let's make use of what we have. Let's stay away from how we're going to get there and focus on what it's going to look like when we do get there. Okay, so simple versus complex. We talk about pooling when we are putting data together from multiple studies. But pool is a statistical term used when we talk about, we will talk about different pools for different kinds of analyses that we do, depending on the study or the phase of the study, that sort of thing. So a whole study may be included in a pool, but another study not included in that pool, or part of that study might be included in a pool and not. So we have different pooling needs within integration that's different than an individual study. So when we start trying to put this data together, we have to think about the different kinds of pools that we might need to assemble. So there's an example here at the bottom of the slide with a double-blind study and an open-label study. So if you try to put that integration together, you may end up talking about different analysis pools. One that has the double-blind information in it, maybe one that has all the information in it from both the double-blind and the open-label when they were active. So we can have different kinds of pools that we use. So it's something we really had to keep in mind as we were putting this integration together. And that kind of got us to what's a simple integration versus a complex one. So when you have multiple pools, you're talking about a more complex integration. When you have exactly one pool, then you kind of don't even use the word pool. Then it's just that's more simple. The other complexity is when you can have subjects enrolled in more than one study. So if you have subjects in exactly one study, then your integration is much more simple. But when you have maybe an indication that doesn't have that many people who have the disease, and so you let subjects enroll in different of your own studies over the course of time, then it gets a lot more complex to pool that information together because you don't have treatment naive subjects when they get into their third and fourth studies. Things are different. Okay? So it gets more complex, and then you have to get into more pools again. So that was sort of how we decided on the simple versus the complex. So simple being each subject's enrolled in one study. They're not enrolled in multiple studies, and they only have the one pool that we have to worry about for analysis. Yeah.
Just to be clear, if you're talking about the scenario on the previous slide with the open label extension, in your terminology, are you thinking of that as the same study or a different study?
Yeah, if the only other study a subject is enrolled in is an open label extension, and you're kind of treating that as sort of an additional treatment period, then you might be talking about simple. If you're treating it as a different thing, you know, it could get in, that's a gray area, whether an open label extension is considered a separate study or not. Sometimes we really just think of it as an additional treatment period within a study. We can treat it like that for analysis. And then that's more simple. Okay, so let's talk about simple first. All right, and honestly, this is where I have typically landed when I've done integration. I've landed in simple. So my goal when I was on the integration team was to make sure they didn't muck up simple, because that's all I ever did. Okay, so let's see if I was successful. All right, simple integration, we found that we could use the existing structures that the atom already had. We had an ADSL, a BDS, and an occurrence OC-CDS structure that worked really well for this simple case with one study, each subject had only one study that they were enrolled in, or potentially a extension study that could kind of be tacked on. And we weren't dealing with multiple pools. We could make use of the existing structures. And for those of you in the room, if you've been doing integrations like this, this is probably what you've been doing all along, is taking advantage of these 3 different classes we already had and using those for your analysis. And you may have run into some challenges, okay? So some differences between an individual study's ADSL, for example, and an integrated ADSL is you have more than one study ID in the data set. So an individual study, you only have one study ID. You could kind of ignore study ID. It's there, so we can populate it and have it be different as you look at all the different subjects. So that's a difference. The other difference is the subject level population flags. So in an individual study, those population flags cannot be missing, right? That's one of the rules. You have to have your population flags be non-missing within an individual study. But that rule doesn't really apply when we're talking about integration, now does it? Because a randomized flag doesn't make sense for a study that wasn't randomized. Does everybody get a no? So you don't typically use those flags the same way when you're talking about integration. So in this case, it's perfectly legal to have missing values in a study level population flag, because for that study, that flag didn't apply. So it would be populated by study. If it applies to the study, then everybody's a yes or a no. If it doesn't apply to the study, everybody's a missing. Okay? So that's a difference, but very minor stuff. Now, I grant you, Pinnacle 21's probably going to yell if you do this, right? But Pinnacle 21 doesn't own the world, and we have a way to say in our reviewer's guide, there's a reason why it looks like this, and this is your opportunity to explain that. It's a perfectly reasonable explanation, and no one's going to give you any grief because it doesn't make sense to try to populate those things when they don't apply to a study, okay? So simple integration, this works really nice. Couple of different rules, and we're talking internally about how do we apply those rules and how do we invent some rules that apply to integration when we're using these same standards. Okay, so simple integration example is an ISE, Integrated Safety of Efficacy. So here, what we're doing is combining 3 phase 3 studies together. They had similar designs, enrollments. None of the subjects enrolled in multiple studies. We only have the one pool, so it's simple. And we're just basically going to stack the data together. So this is kind of assuming that you've got some atom data that's already well structured and similar across those studies so that we can stack them on top of each other. And here we're going to see where we've got an ADSL data set and an ADEFF data set in each one of those 3 studis so that we can stack them on top of each other and now have an ADSL data set that has all 3 studies in it and an ADEFF data set that has all 3 studies in it. Okay. So we'll have the integrated version. Now this is a very simple example. I've never seen this work in real life, that you could design 3 studies to do 3 different sets of, and actually stack them on top of each other. Boom, you're done. We'd all be out of a job, right? The programmers in their room. But in theory, this could happen. And you know, in real life, you may have to do a little tweaking, but you could get it to be fairly simple. You may be maybe pulling from SDTM data instead of ADAM data. That's maybe a more likely scenario, but hey, you know, perfect world, this is a nice thing to have happen. All right, so that's simple. So that was my goal. I wanted to make sure simple stayed simple. Now let's talk about complex, all right? So again, complex is when you're enrolled in more than one study and you have multiple pools. So we need a way to handle that. And we'll start talking about ADSL and then we'll continue on to the other structures. We need a way to handle this. So we started playing around with ideas. Okay, we got two studies, a double-blind and an open label, and two pools, double-blind and active drug, and subjects maybe in one or more of those studies. What are we going to do with things in ADSL like treatment start date and intent to treat flag and safety flag and age? If they're involved in two different studies and they're started a couple of years apart in time, this subject's gotten older. So the age he was in the first study and the age he was in the second study are not the same thing. So you can see how, and then you start adding more studies and then more subjects going across these studies, and it starts to really bubble up. We're having trouble figuring out what to do with all these things. So we thought about this. This was our original idea. We started with our standard ADSL variables that were across all the studies. And then we had specific variables for the double blind. That's the middle column. And then the far right column are the specific variables for the active drug. Okay, so our ADSL structured, you know, one record per subject and things kind of going across. We started struggling with this one though, because we had variable naming convention issues. You know, yeah, you could take something like age and have age 1, age 2, age 3, age 4 going across. But what are you going to do with things like the Treat-O-1P? That already has a 1 in it. Or the treatment start date, you know, Treat-O-1, the start date for that. You're maxing out your 8-character limit, and now you need to put numbers on them to try to get them to go across. So, and they start making less and less sense as you start trying to stick all those extra numbers in there. And we could see right away that this was going to be a problem, and we were going to probably need 2-digit numbers because integrations often will combine more than 9 studies together. You could get some crazy integrations. So things are going to get really wide, really fast, and we're going to have some crazy numbering schemes in there and variable names that don't make much sense. So this really didn't stand the test. This would have been a simple way to do this, but it really didn't stand the test of being a true standard. So what we ended up with instead is the iADSL structure, where we started again with the common things, the standard ADSL variables that were overall across all the studies. We added pool as a variable so that we would know which pool we were talking about. And in that standard stuff that goes across all the studies, we call that the overall pool. Then we add rows. My God, we're adding rows to ADSL. Okay, I know, it's scary. But we added rows for the pools that that subject belongs to. So if a subject belongs in both the double blind and the active drug pool, they're going to have those 2 more rows added. So the ADSL is actually 1 record per subject per pool plus an overall. And the overall gives us this nice feature where you can still, if you subset to overall, you're back to 1 record per subject. So boom, you can have your 1 record per subject that everybody's very comfortable with. It just doesn't have all of the detail that you need for those individual pools. All that detail is in those additional records that you're going to need for all those other analyses as well. Question?
So for the overall, what data value do you use? Because they will have 2 different values for each.
Okay, so for the overall, if I scroll back up here, I think I scrolled too far. No, I scrolled down. There we go. For the overall, you only have the variables in there, that you populate the variables in there that apply overall. So some of these variables are not going to apply overall. If you have different ages, you may care about in the overall the first age. You're going to have to define what those are. Some of them you're not going to, maybe you have the first treatment. And some of them you're just, they just don't make sense when you talk about overall versus the double blind or the treatment pool, because some things only make sense when you're talking about the individual pool. And we're going to see an example later on, and maybe that'll help. Okay. So you only populate the variables that you need. You only populate the rows that you need. So just that subject, there's a subject that's in both of those studies. They're going to have both of those rows. But if you're only in one study, you don't need both of those rows. And if you've got 5 studies that you're combining together and you're only in 2 of them, then you only need the 2 rows. So you create the rows that you need, and you create the columns and populate those columns as you need them. Okay. So here's an example of how we kind of put that together. So we've got 3 subjects here, and we've got 4 different studies. A phase 1 dose escalation study, a phase 2 proof of concept, the pivotal study, and the open label extension. So no one was in all 3 studies, or all 4 studies, excuse me. The first subject was only in the first study, the phase 1. And then the second subject was in the phase 2 proof of concept, one of the phase 3 studies, there's 2 different ones in there, and the extension. The third subject was in the phase 2 proof of concept, the other phase 3 study, and the open label extension. So here was an example that we started with. This is simplified for the slides. The example in the document is a little more robust, but it does show that we have this, it's still an interesting scenario. Okay, so subjects in multiple studies, and how do we deal with that? Okay, and this is how we dealt with it. Okay, so here's our pools. We got 3 different pools. The overall pool has all the studies. The second pool is the pivotal pool. So that includes just the phase 3 studies. And then the third pool is the comparison pool. So all the studies that had a comparator arm to them. And so that's just the phase 2 study and the 2 phase 3s. It's not the open label and not the phase 1 study. So 3 different pools. We got our overall, our pivotal, and our comparison. All right. And here's how the data, some of the data might look for those 3 subjects. The first subject is the one in bold up there now. He was the guy that was just in the phase 1 study. So he only has the 1 row, the overall row. The phase 1 study is not part of the comparator pool or the pivotal pool, so he's not involved in either of those. Right, so 1 row overall, that's it. Second subject was in the phase 2 study and 1 of the phase 3s and the open label. So he's in 2 pools, or yeah, the overall pool plus the pivotal and the comparator pool. So he's got 3 records, right? And then the third subject is in the overall, the pivotal, and because he was in the other phase 3 study, he's in the comparator study there as well. So we've got, what's that, 7 rows to cover 3 subjects, right? And again, if all you need is the 1 record per subject version, you subset to get just the overall pool, which in this case is pool number 1. Or you could do pool equals overall if you want to spell out the words. I'm a programmer, I want to do pool n equals 1 because I like shortcuts. So we can get it down to 1 record per subject if that's all you need. Right? And if all you need is just the pivotal, you subset by pool 2. And if all you need is just the comparison, you subset by pool 3. So it's a holding place for all this ADSL data, but you're probably never going to use it all together at one time. You're going to use one of the pools to do your analysis. Does that make sense? All right. That's what we came up with for ADSL. For the complicated case. Question?
Maybe I'm jumping ahead here a little bit, but like it seems like you can run this new comparison tool in terms of what is being populated in that row, because you sort of have data from different studies. Right?
So in the comparison pool, we're going to run into trouble about what's being populated in what?
Well, in some of the columns.
Yeah.
Okay.
So you're going to have specifications on how to populate those things. And, you know, your SAP is going to have to tell you. So this one, it's probably the treat 01 start date was the first treatment for the first study. And then you're going to need treatments for those other studies, and how you define what that is. We're not going to tell you that. You know, your statistician is going to tell you that. It's how you're going to analyze that. Yeah. Question?
Yeah, I got one from
Okay.
the webinar, specifically about the slides.
Okay.
I wanted to sneak it in.
Yeah.
So the studies variable or column, would that replace a regular study ID or multiple study ID column?
Yeah, so the studies column is in there to show you which specific studies the subject was included in. That's hard to capture with our study ID that we have now. So because you're combining studies together. So study ID still works really well in the simple case when each subject's in exactly one study, but it doesn't work well here. If we tried to use it here, we'd have to have one row per subject per study, and then that's not analysis ready. So we ended up with pools, but then pools didn't tell us what studies the subject was in, and maybe we want that information. So we really just created a text open field here, studies, where we tack them all together. There's no like requirement, it has to be a comma and a space or whatever. You just need a way to describe what studies that subject was in. It's more for human readability than anything else to give you that traceability. Question?
So for row 4,
Mm-hmm.
the subject has 2 studies. What do you use for the first dose?
What do you use for the first dose date when the subject has 2 studies, like on row 4? Your statistician would have to tell you what to use. I'm not going to tell you. Yeah. So yeah. Question?
Maybe I missed it. Is there standard terminology for the pool variable?
The only standard terminology for the pool variable is the word overall. All the rest are up to you to determine. Yeah. Question?
So I say this variable in this screen that the TRT01P, right? And TRT01 start date and date. It is associated with the pool defined across studies.
Yeah, so what you see here in treat 01P, treat 01 start date, treat 01 end date, these are standard ADSL variables, but the way they're being used here in the integrated ADSL is they're potentially different as you look at different rows within the subject. So if you look at the last set of rows, they're the highlighted ones. Row 5 has a treat 01 start date of August 29th, but row 6 has a treat 01 start date of September 6th. That's because row 5 includes the 201 study, the phase 2 study, which started before row 6's only study, which was 302, which started, that subject started that study in September of 2001. So your SAP is going to tell you how to define all those things, but this gives you a way, because when you do your analysis, if all you're analyzing are the pivotal ones, then you care about the fact that they didn't start until September. But if what you're analyzing is all the comparators, you care about the first one potentially of when they started. So
it look like this for the first phase of the treatment, you cover 3 studies for some prophylaxis. So if that different treatment in different study used, how do you differentiate at this case?
So treat 01P is what they received in the first study, or in the first treatment period, however you've defined that. So what they received between September 6, 2001 and April 27, 2002 is what is in treat 01P. And then on the third, or on the bottom row, row 7, between August 29, 2000 and September 24th there may be multiple things that go in there. So it may not be just Treat-O-1P. Maybe what you have is a Treat-O-1 and a Treat-O-2. So that's all dependent on your analysis needs. You have the variables, and it's up to your statistician to determine what goes into them.
So for this case, if they are across 3 studies, I assume that you will say 3 period treatment and 3.
There could very well be lots more rows out there. And if you look at the document that's out there for public review right now, you'll see lots more data. We got this much real estate on the slide. So we only gave you a snippet on the slide of what's out there. So I encourage you with questions, take a look at the document, because this is just a snippet of the document. It's got a lot more detail in it. Question?
So you said the only standard for pool is overall, but what if your analysis doesn't have an overall?
We still want you to have the overall row, even if your analysis doesn't call for an overall pool, because that gives you that opportunity to subset it down to 1 record per subject. You may not populate all the variables on that row because you just don't have a definition for how to populate them, a need for why you need to populate them, but we have you create the overall pool. I got to keep moving on here so we can have more questions later. But we got to apply this IADSL to other places like BDS and OCCDS, okay? Because now we got multiple records per subject in ADSL. How is that going to drive what happens in BDS and OCCDS? Okay, so this is what happens. We end up potentially with multiple records for the overall pool, the double-blind pool, the active drug pool in the same data set, right? So what that looks like, well, actually a little more detail first. So we end up creating records for each of the pools that we need. So some records don't apply to some pools. That's okay. Don't manufacture data. If you need that data for that pool, then you populate it. If you don't populate it. You keep the records that are relevant for that pool, right? Having it in this more vertical structure is going to be advantageous, which we saw with ADSL, and it also shows up here, because things are different for different pools as you move through them. We just saw treatment start dates were different in ADSL. That's going to affect things like study day when we get into OCCDS and BDS, because we use treatment start date usually to determine study day. So if we have a different treatment start date, we're going to have a different study day in 2 different pools. So this allows you to have that flexibility. You can think about this, I go back to this slide, you can think about this as like having 3 separate data sets, all right? So the first data set has everybody across all the pools and the basic information. The second data set is just your double-blind pool, and it has just what you need for that. And your third data set is just your active drug pool. The only difference is we put them all together in one data set so that you have all of that handy, and you can just use where clauses when you need to pull out the specific information for your analysis. Okay. So let's look at an example of an IBDS, an integrated BDS. Okay, and I'm going to move through it like the last time. So here we're just looking at subject 3, right? I think this was our second subject actually. And we're moving through this particular subject's lab data. So in the pivotal study, there are 2 records, but in the comparison study, there are 4 records. How'd that happen? Well, because there's 2 different input studies to the for the comparison, because they were in study 201 and study 301. So they have 2 records coming from 201 and 2 records coming from 301, where the pivotal only had 301. So you only have those 2 records. So see, again, it's like having 2 different data sets stacked on top of each other. And you can subset to pull out the pivotal pool or the comparison pool. All right. Let me run through some facts, and then I'll open it up to some more questions. So Is the ADSL class required? And this is a no, because in a lot of our integrations, we don't have these complicated multiple pools, subjects in multiple studies. So you don't need to use the integrated version if the regular version applies. Okay, so we did the simple piece first. If that meets your needs, go for it. Use the simple stuff, all right? If it doesn't meet your needs, then you want to use the integrated classes. Additional ADSL class support for integration. At this point, no. But like I said, we do want to create some rules, some ways of checking your data, because the integrated version of ADSL is going to be different with different study IDs and potentially missing flags, population flags. That's different from the regular ADSL. So we may end up doing a little tweaking on rules for that. Is the dataset name still ADSL? Yes. We don't put an I in front of the dataset name. There's an I in front of the class, but not in front of the dataset name, because the AD is the reserved prefix for all ADAM datasets. So we have to have that AD at the beginning. So we're just using ADSL. The structure, the class that applies to it may be different, but it is still called ADSL. Compliance checks validation rules. So there will be compliance checks and validation rules written for this when it comes out. There is not right now, because it's out for public review. We're not going to write a bunch of rules on something and then potentially change it. Okay, so we will write some rules as we finalize this. What if use subject ID wasn't correct and unique across studies? Adam is not getting involved in that one. If somewhere up the line, upstream, the work to make sure that the use subject ID is correct and individual subject is properly identified across all those studies, if that didn't happen, we're not going to try to fix that for you. You're going to have to figure out how to fix that yourself, because it should have been correct up here, and Adam doesn't fix problems that are up here. Adam fixes stuff down here, all right? So you're going to have to figure that one out. We're not touching it. Re-enrollers in different studies or other complicated scenarios, I'm going to point you back to the document. Okay, so it does talk about, the integration document does talk about this stuff. We didn't cover it in the slides. Dataset size. Well, you can split your datasets, ADAM datasets, by anything. If your dataset gets really big, you can split it up. And pool is a really nice way to potentially split up a dataset, because as we were just talking about, it's really kind of like multiple datasets stacked on top of each other. So it makes it a nice variable to split by if your dataset gets big. In fact, you could just create them that way. Nobody's going to stop you from doing that either. Linda.
So what do you call the dataset if you split it? Because you've got to have an ADSL.
Yeah, so if you split your dataset, then when you go to your submission and your define, you need an ADSL that would have to live in that primary dataset submission folder, but then you have a split folder. And in the split folder, you'd have whatever you call those different splits. Okay, so you still need that ADSL, that's our rule, but you can split all the rest of the stuff. And then can you implement these structures now? Well, this document is out for public review. I'm taking my CDISC hat off now. Everybody see that? No CDISC hat on. Putting my, I work in a consulting company hat on, okay? So I will say that if you are working on a standalone study, this is a decent model to follow to develop a study for submission, because we don't have a standard for integration. But if you are trying to develop internal company standards, I do not recommend you work with a draft version of any document, because things can change based on what you guys say. I'll give you an example. The ADAE document went out for public review years ago, and the variable names for dates, we had somebody comment on them that why are they named this and shouldn't they be named that, and they had a really good argument. We talked about it internally and we agreed with the reviewer and changed the names of the variables based on that comment. People yelled at us because the variable names were different in the final version than they were in the draft. We improved it based on your feedback. So that stuff happens all the time. That's why we send things out for public review, so that we can get your feedback. So do not base standards around this stuff that you're seeing here and the draft document that's out there. But if you're doing a one-off study, sure, why not? Because there's no standard out there, so you might as well do this, okay? But that was my non-CDISC hat, just to be clear. Question? I am not going to guarantee you're not going to get questions from a reviewer, but I'm saying in the absence of anything else, why not give this a shot? Yeah, there is no standard. So there's no standard out there for integration. You could do whatever you want to right now. This is an option. Okay, so conclusion, draft out for public review right now, closes May 21st. You still have almost a month. So if you've not taken a look at it, those of you that are asking lots of questions, please go out to the CDISC website. If you don't already have a username, get out there, get your username, pull this out, read through it, make comments. We want to hear from you, right? Acknowledgements. So she acknowledged Wayne, who wrote the initial version of the slides. I'm going to acknowledge Deb, who presented these slides originally, because her name's on the front of the slide. But then the entire Atom integration team helped put all of the meat together that's behind all this. So all the examples and all that stuff. All right. So I am now open for questions if I have time. How are we doing on time? We are short on time. So, all right. So are we going to do questions or no? Okay. So, Linda, your hand's up.
Yeah, what's the timeline for this being in the catalog?
The timeline for this being in the catalog. Well, so it's highly unlikely that this is going to get comments in resolved and the document finalized with the rules that have to go out with it by this fall's timeline. So it's very likely that this document will not come out as final until fall of next year.
Is this something that would actually go in the catalog?
And then, I think by catalog you mean the FDA, Data Standards Catalog, and then FDA is going to have to spend some time playing with it before they decide whether or not they want it this way.
Will this constitute a new version of the Atom standard?
The Atom model?
Yeah.
Yeah. So I'm going to talk more about that one later. So hold off on that question because that's part of my later slides, whether this is going to make for a new model. But, you know, none of us here can speak for the FDA. I can say that in the past, FDA has adopted every CDISC standard that we've thrown at them, but it's just taken them a while to do it. So will they put this in the catalog? Probably eventually. I can't give you an estimate of when that would happen, but yeah, they're aware of this. We've asked them to do a review as well. So we have contacted people in different parts of the agency to get involved. I haven't seen any comments back from them yet, but yeah, I'm hoping it'll get in catalog. Sure. Okay. Should we break? One more online? Okay.
So this question asks, it seems like the pool variable is sort of similar to base type, but able to go across different studies as opposed to just within a study. Is that a useful way of thinking about it?
So is pool, yeah, you know, I think it is kind of like base type where if you didn't have, if you wanted to split it out into separate data sets, then you wouldn't really have the pool. The pool would be part of the data set name. And base type's the same way. If you want to split out your baselines into different data sets, then you don't have to have a base type. So yeah, I think that is a really nice way to think about it. If you understand the concept of base type, you can apply that to pool. So thank you, whoever that was. All right, let's reshuffle ourselves for the last part of the day. Okay. All right, thanks. So hi, Sally. John has me introducing you.
Oh, great. Thank you, Sandra.
So I've known Sally for a long time. She is actually now working for CDISC. So a lot of you that have known Sally like me may not realize she is now an employee of CDISC. She doesn't just do this as a volunteer like the rest of us do. So she's been on the Define team, leading the Define team, training on the Define forever. Well, no, you're not that old. Yeah, so I've run into her at many of the meetings working on Define, and she and I have worked together on some of the things that kind of cross over between Atom and Define. So I see the subclass on the slide here, so that's something that she worked with the Atom team to develop. So it's been really, really great having Sally now being part of CDISC, because she understands the Atom material that I work with all the time and can kind of be a voice for Atom, because we don't have an Atom employee. So Sally is our Atom employee over at CDISC. Thank you, Sally.
I'm trying.
So she knows this Define stuff really well, and as I mentioned, she's a teacher. She does the training on Define. So if you ever take a Define class from CDISC, you are likely to run into her. She's not the only Define teacher, but she's a prevalent one. And one of these days, I'm going to take Sally's class, just so I can. Great. So I'm going to let Sally take it from here. Welcome, Sally, to our little group here in South San Francisco.
Oh, well, great. It's nice to be there, at least virtually, and nice to have a chance to talk about Define-XML version 2.1. I guess I won't be able to see if I ask you how many questions, but I'm hoping that, well, I guess how many of you have know about Define 2.1? Know this is a new version of Define coming very soon?
We have...
Any hands? All the hands? No?
There's a few.
Okay. Okay. Well, we've been at this for almost as long as Fred has been working on SDTM 3.3. Maybe not quite so long, but and it's nowhere near as big of a change, but there we have it. So we have Define 2.1, and really the big changes are in three main areas. We've added metadata to let you identify the standards you're working with in a particular Define-XML document instance. We've made some changes to the how origin is represented for SDTM data, and re-implemented the class, what used to be an attribute, to allow for subclass. So I'll just talk about those. I won't take long because I know we have some other, perhaps more interesting topics to talk about in a bit. Let's see. Okay. Displays. Can I move it? Yes. There we go. Okay. Well, before as I start, I'm going to talk first about this standards identification change. But I wanted to, if you can see up here, you might, and if you're a frequent Define user, you may notice that the style sheet looks a little bit different. So I wanted to just make you all aware of that there is a new XSL style sheet that was developed through the Fuse CSS working group. And that's kind of become the standard style sheet for the Define-XML. It's actually works with Define 2.0 and 2.1. And if you now download the Define-XML kit from the CDISC website, you'll get this updated style sheet included. So that's kind of a nice thing. I won't go into all the details about it, but it does have a lot of nice new features, including options for how you display the value level metadata, and it actually does, I think, a nicer job of showing the information without showing you quite so much of the insides of the Define. So I think you'll like it when you start using it. I know I do. Anyway, so as I said, one of the things that people kept asking us for was the ability to declare within your Define-XML which standards you're working with. And I mean, often in a very nice simple case, you've just got vanilla SDTM-IG and no devices, no associated persons, nothing, and so it's very simple. But in many cases, things are a little bit more complicated. In this case, I'm not sure this was how real life this could be, but it's a case where the study started a while earlier, and then it moved to a later version. And by the way, it uses devices, and so we wanted to be able to make that all clear right at the top of the define. Also, in some cases, you may end up with more than one version of control terminology in play, particularly if you're working with Atom and you have some Atom terminology you're using and you have some SDTM terminology you're using. There are cases where that might be useful. And so it's used to identify both the content standards used by the dataset definitions and all of the controlled terminology versions that are referenced anywhere in the study described by the define. And then each dataset definition identifies the standard that it follows. Now, if you have a non-standard dataset, it defines itself as being non-standard, and so so every single dataset is now tagged with a version identifier or an indication that it's not standard. So I can take questions now, or I can take them at the end. Maybe I'll just keep going. So the other kind of significant change involves the origin metadata for SDTM, where we've kind of broken out or deconstructed the origin and now separately identify the source, that is the party who's responsible for generating the data, as separate from the type. So in this case, we've got several variables that are the responsibility of the sponsor, and some that are collected through an investigator site. And that's just an additional piece of metadata that gets added to for each variable when you define the origin. The other thing we allow now is allowing you to specify more than one origin in cases where you more than one source for a particular variable values in a data set, and particularly in cases where it's not really straightforward or easy to describe as part of your value level metadata. And so if you do provide multiple origins, they'll both be listed in the style sheet, as you can see in the example here. And then last but not least, certainly for our Atom friends, is again, kind of making the class metadata a little bit more complicated, but then again, may be more powerful by allowing the addition of a subclass. And so to do that, previously, if any of you know about Define-XML coding, you know that previously it was an attribute defined as part of the item group def element, and now we've to be able to get the subclass and to be able to we've made that quite flexible so that in years to come we can have a more complicated model, I guess, and so that now def class is a child, is an element, and the subclass is a child of the class. And you can have multiples and you can combine them according to the schema, but so far it's not really used anywhere, although I think Atom is planning to use it with some of their newer publications. And so that is available. And for now, you can just convert the class values you've been using all along. But know that in the future you'll be able to use a subclass if that's helpful to you in defining new SDTM models or in Atom. And so far, again, we're working with the control terminology to develop control terminology for subclass code lists, but that isn't quite ready yet. So that's it on Define-XML. Are there any questions?
Hang on.
When?
When? Yeah. Sandra's question is, when is this going to be released?
Oh, gee, I wish. Pretty soon. I think we're in the final stages of the CDISC publication process. Although every week I keep saying that, and every week it seems like there's something slightly more that we still need to do. But pretty soon. I'm hoping by let's say the end of May, maybe, if we're lucky.
Well, that's soon.
Oh yeah, no, it should be soon. It should be. I mean, yeah.
And then the next question, which you absolutely can't answer, is how long is it going to take the FDA to accept this? I think
you got that straight.
It took them 2 years or something like that, didn't it?
Yeah, I think as with all the standards, once we release it as a production standard, they can start trying it out and kicking the tires. And
so I have a couple of questions for you. One is the style sheet for 2.1 backwardly compatible if you feed 2.0 stuff into it? Is it going to display correctly?
Yes. It's actually it was designed with both of them in mind, because it was kind of put together in the long interval during which we're developing 2.1, and so it does handle the 2.1 stuff nicely, but it is absolutely the same one that is available for 2.0, and the intent is to keep there being just one.
Okay, good. That's good to know. And the other question is seriously nerdy. The ability then to have multiple origins, is that going to change the rule that if you have multiple origins at the value level, that you leave origin empty at the variable level, for example?
You could, no, yes, it will change the rules. So they'll have to be a little bit more flexible there, because I think you could do it either way in that case. So, yeah, we haven't finished with the rules yet, so we'll probably be thinking through some of that.
Okay. Any other question? Okay, we got a question from.
This is actually just for me. Hey, so my question is, are the new attributes like subclass, standard version, all that, are those going to be required in the XML, or are they optional?
The standard version is going to be required, and one of the things we've done is we've added an attribute actually at the very top level that lets us kind of define rules for submission use cases versus any other use cases. But yes, the intent is to make them required for submission use cases. And we went around and around and around on that. But yes.
So Sally, I think it's time to move on to the CDISC news.
Will you, can I tell you to click? It's got to build, so do you want me to be a presenter, or do you want to? Oh, thanks, good, thanks. Okay, well, I think, again, I can't see the hands. Hopefully the news has gotten out that what we used to call share is now CDISC library. So it is a whole new version of whole new product, really. It's a new platform based on the semantic technology stack. Uses linked metadata model, which is much better suited for ongoing support and development of our standards. The The API has been expanded, and I think is much more usable than it was for Share 1. And it's been in general release for a couple of weeks now, 3 weeks, I guess. And if the next build, I think, starts showing you what's in there. Yeah. So for content, we've loaded in all of the CDISC model and all the published IGs, and again, many of the SDTM models and the IG 3.3 is in there as are the associated persons, medical devices, PGX. And the next click will give you the ATOM. There is both versions of the ATOM IG and the published versions of the OCCDS, the time to events, and the ADAE. Plus all of the controlled terminology for all of these, all of the standards, basically all the controlled terminology publications since I think like 2014 or so are in there currently. And really, that's all I have to say about that. Although, you know, if you are a member of CDISC, I'd really encourage you to sign up for an account. You can go to the library page on the CDISC webpage. And also, I don't know if there was quite a good webinar about a month or so, maybe a month and a half ago, to introduce the upgrade. And if you haven't seen that, I'd encourage you to look at that also from the CDISC website and run through that, because it really is quite good. So I think it's going to be a wonderful thing. I'm really excited to see Ram's presentation, because a lot of what they're doing at Roche is what we want to be doing with our own standards, and so that's going to be enabled by having us all use the library on a regular basis.
And one of the things that I noted when Ram was talking was that they're using JSON files, JavaScript is their part of their, and so is CDISC library. And I think the last time I talked about this, maybe I heard this wrong, but I thought I heard that it doesn't currently, at least, export spreadsheets like everyone was used to with Share, but rather JSON files, depending on what kind of a query you would give.
Yes, right now it supports JSON, and actually there's sort of a kind of a vanilla XML that kind of mirrors the JSON. And you're right. So far, there's no CSVs or Excel coming out, but we are working on that for our next update, which I hope will be in a couple of months. I don't know what I'm supposed to say about that as a release date, but we are working on a new version that will have the Excel. Of course, the old Excel files are up there, so you haven't actually lost anything, but you can't get them out. You can't get them out for free. You can, of course, do your own programming with the JSON and make an Excel if you want to do that. But yeah.
Okay.
Yeah.
So should we turn over to Sandra? Okay. Let's go back to, there we go.
All right. Thank you. I'm going to buzz through some ATOM updates here. It's been a while since we met, and I thought it was important to kind of convey, what ATOM's been up to. So I'm going to walk through one document that was recently published and 12 documents that we have in various stages of development. So we got a lot going on the ATOM team. So the one that was recently published, does everybody know that we have new conformance rules out there? So it's called version 2.0. We did a major update. It was 1 point something before that. It was 1.4. We upped it to version 2 because the layout of it looks different. We now have checks for TalentMine version 1.0, 1.1, and OCCDS version 1.0, and we have like a column in there that says which version you're looking at. So a lot of the checks are kind of paired, where a 1.0 and a 1.1 check are basically doing the same thing, but they're in their own row so that you can subset to get just the standard that you are interested in. So interesting to note, one of the sub-team leads, a sub-team co-lead for the conformance rules document, just started working for Pinnacle 21. So hopefully that'll make the rules consistent between what the Adam team says and what Pinnacle 21 does, right? It's the same guy. So yeah, so we got that going on. So that's new. If you've not downloaded that yet, you know, we're excited. It came out in March. In development, just in general, there is a screen on the CDISC website where you can find out all the standards that are in development and, you know, a little bit of information about each one of them. So I just dumped a couple of pictures, so it's actually a very tall screen, so the stuff on the right is actually underneath what you're seeing on the left, if that makes sense, just so I could get it on one page. The Adam stuff, because we're alphabetically at the beginning, gets to be at the top, so it makes it really easy to find Adam if that's what you care about. You can scroll down and you can see the SDTM stuff. You can see the data exchange, so you can see, you know, Sally's Define 2.1 is in there. So you can see all this stuff, and then down below are TAUG's therapeutic area user guides, which I didn't cover. But in general, there's all that out there. I'm going to focus on the Adam stuff, all right? So soon to be finalized documents, we have an Adam version 2.1 considerations. This is a one-pager that we're tacking on to the Adam model document, version 2.1, because that model document came out in 2009. Which is like a lifetime ago in terms of standards, right? And most of the stuff in the document still applies exactly as it is, but we want to inform you about a couple of things that aren't mentioned in there. So there's no mention of OCCDS because OCCDS came out afterwards. No mention of more current versions of Define-XML. All the examples look like Define-1, right? And there's a table in the IG that kind of lays out which standards which documents apply to which versions of the IG, and that's a helpful thing. So it's just a little one-page document. I think it's going to be a PDF that's going to be attached to the model document whenever you download it. So that's new. Adam IG version 1.2 and the Adam Geriatric Depression Scale questionnaire, the Adam version of that questionnaire, those two things are imminent. So they're in, I think the GDS is in publishing, and Adam IG version 1.2 is in copy editing just before publishing. So sign-offs have been done, and it's just a matter of going through the CDISC stuff. I'm estimating summer for both of those, so we'll get to see those soon. So those are coming out. It's very exciting. In public review right now, we already talked about this one. So the integration, we talked about this one earlier, so I'm not going to spend any more time on that. It's going on through May 21st. Preparing for public review. We have an Adam IG for medical devices and an OCCDS version 1.1 that have been through internal reviews and public reviews, or preparing for public reviews now. So medical devices is with copy editing, and OCCDS version 1.1 is still in our internal CDISC review. So those things are coming soon for public review. You can expect to see that. You can give us comments again. Then we have this other stuff that's in development. So non-compartmental analysis or PK data, traceability examples, just a whole slew. I think there's almost 20 examples of traceability. Oncology, things that apply specifically to oncology, stuff that don't in other documents. Questionnaires, so they're just going to keep grinding out these questionnaire supplements for Adam. More conformance rules for every one of these documents you saw. We're going to have conformance rules that go with them. When you're creating a new standard, you need conformance rules. And then Adam 3.0. This is a huge undertaking for our team. The goal here is to take all these documents, all these little documents that we've been talking about, and get them under one roof. instead of trying to refer back to this document and refer back to that document, we're trying to pull it all together. This is a big undertaking. It's going to take us a while to get this one accomplished. It's part of the reason why we had that little one-page snippet go out that attached to the current model document, because it's going to be a while. Right? So that's what we got going on the Atom team. We just did this slide, so I can skip over that. And you want to talk about TALS. You're up next, John, right? Or do you want me to answer questions on my stuff first?
I don't think we have time.
Okay, go to TALS.
So TOGS isn't me, it's Sally, I think.
You're back to Sally?
Yeah, Sally, are we back to you for TOGS?
Oh, you muted Sally.
You have been muted. Okay, now you're back.
I'm muted. Okay. Yeah, this is just a subset of the page that Sandra was showing, and as she said, this is actually available on the CDISC website. You can find it pretty easily, and it just indicates the ones that are yellow are the ones that are some way along the being done, and the ones that are green are actually, in these cases, all pretty recently released. We just, CDAD is just about, I think it may be even out this week. I mean, they're really close on that one. The lung cancer therapeutic area guide was, I think, announced a week or two ago. I'm not, the nutrition therapy one, I don't know what's happening with that. I haven't had much to do with that one at all. And the traditional Chinese medicine is also preparing for publication. This is kind of the coronary artery disease part of Chinese traditional medicine. Some of these others are like the psoriasis and the acute kidney injury, congestive heart failure, diabetes type 1, Crohn's disease are all in various stages of progress. There's a large effort on the type 1 diabetes project. There's kind of two general projects with each with a number of modules associated with them. So that is, the whole thing will take a while to get done, but it is that you will see public review opportunities for subsets of those as they come along. And yeah, Crohn's disease is just starting, I think. So that's what we have.
Okay. Let's see. So Sally, any other sort of news from CDISC that you want to sort of throw out there before we move on to other stuff?
Not a whole lot. As you know, the European interchange is, I think, in a week or two, and so there will be a number of news items associated with that, including I think the final report from the Blue Ribbon Commission and sort of a new 3-year strategic plan. So I won't scoop anybody on that, but stay tuned for that in the next couple of weeks.
Actually, I have a question. For those TA guidance, are they just for SDTM or for both SDTM and Atom?
Actually, all of them we try to do CDASC, SDTM, and Atom where there is something interesting to show.
Okay. Thank you.
Okay. So let's, thank you. Thank you, Sally. Don't go away, because you're still going to be more stuff for you here. One of the other topics that I put on the menu here was it's about the technical rejection criteria. And I don't know if this is on really very many people's radar screens right now, but there are some things in the technical rejection criteria that are very concerning to me, and I think that the FDA is not fully aware of how much concern they should have about some of the stuff in there. So what I did was I just grabbed a few slides out of a presentation that that a couple of the FDA folks did at the Fuse Connect in 2018, where it was Ethan Chen, Lillian Rosario, Ron Fitzmartin, and Ginny Husong, talking about what the implications are of the technical rejection criteria. So just so that everybody understands what these criteria are, what they mean, is that if a submission fails an important technical rejection criterion, these criteria are applied, in principle, are applied as you are trying to upload your submission through the gateway to the FDA, right? That's when the submission is inspected to see whether it meets these criteria. So up until 2017, all of the criteria were about the ECTD XML backbone. So if the gateway looks at the backbone coming through and it goes, you know what, I can't do anything with this because there are errors in the backbone, then it would go, okay, let's just stop here. You can have your submission back. We never saw it, right? So now they've added some more technical rejection criteria, 4 more. And you run the same risk of the FDA saying, oh gosh, we never saw that submission, as you try and upload it through the gateway. They're not yet enforcing these criteria yet. They're not enforcing them yet, but they've announced that on 30 days notice, which I guarantee hardly anybody in the room will ever see, on 30 days notice, they can start enforcing them. The reason they haven't enforced them yet is because they figured out that they'd all be sitting on their thumbs while sponsors, scattered to the winds trying to fix their submissions, because 1/3 of the submissions that they receive would have failed the criteria. And I'm going to just show you why some of them are very hard to deal with. One of the things I'm kind of concerned about is that as of this moment, still today, Pinnacle 21 only evaluates ADAM 1.0. ADAM 1.0, as of the 15th of last month, is no longer a valid standard in the catalog. It has expired.
Oh, that's based on the study start date.
Mm-hmm.
MIG 1.0 for new studies, you can't use it, but you can use it for old studies.
Yeah, if you ask the FDA that question, you will get 3 different answers. So I wouldn't count on that. But the problem with it is that when you say in your Define.XML that you used ADAM 1.0, whether you did or not, that is grounds for rejection of the submission. So that's a conundrum. And it turns out probably that if you really used ADAM 1.1 or 1.2, it's backwardly compatible to 1.0, and so you could call it 1.1 or 1.2, and nobody would argue with you. But here's this tripwire, which is that if you said in the Define.XML that ADAM 1.0 was a standard, that standard is no longer accepted in the data standards catalog. That's grounds for rejection of the submission. The other one that's of really substantial interest to me is the second one, 1734. Trial summary data set must be present for each study in the ECTD. Now, that one, it's pretty easy to create a trial summary data set that all it has is a start date in it. However, that's not the problem. The problem is that the rules for the criteria want to verify that TS data set is really for that study. it's apparently not good enough that it's in the studies folder. Why would you think that the TS data set in that studies folder is not for that study? But what the rules are going to do, as currently written, is look inside the TS data set and see that the study ID in the TS data set is the same as the study tag, the study folder in the ECDD. Now, that's a really obscure technical fact, but it's really important. And the reason it's important is because the study tags in the ECDD backbone, those have to obey certain rules about file name conventions. File names in the ECDD have to be all lowercase, characters, numbers, and hyphens. That's it. Characters, numbers, hyphens, characters, all lowercase. In all of the submissions that I've seen, all of the studies that I've seen submitted, I'm going to say in the last 12 years, none of those study IDs obeyed those rules, because they either had uppercase characters in them, or they had spaces, or they had whatever stuff. And if that were true, after these rules are implemented, then the gateway would look at that and go, oh, this TS data set isn't for this study, and your submission is down the drain, right? So anyway, we're going to have a much more substantive discussion of this in the next meeting. This is just to sort of put it on everybody's radar screen that on 30 days notice, really a lot of bad stuff could start happening. And I don't think it will start happening as the FDA realizes that they've made us a sort of a fundamental error here. But I think you have to be aware of risk management issues with this.
Yeah, so FDA did have a meeting recently where they walked through the technical rejection criteria. It was the last of a bunch of things they were talking about. And they did mention that the error rate has dropped, so that 33% is now down to 20-something, 24%, I think it was. So they are actually looking to turn it on for the end of the year. They also talked about some tweaks they made to those rules. So that may address some of your concerns. And I've been watching their website. I haven't seen the slides posted yet, but maybe when they do get up, we can post them to our link as well. Yeah. So I'm keeping an eye on it.
Okay, good deal. Yeah, and one of the things I mentioned earlier, for those of us who are going to the CSS, the Fuse conference in June, we should get together and go, gang tackle an FDA person and see if we can get some clarification from them and see if we can recruit them to get into this conversation with us.
They don't need to be gang tackled. They're very happy to have these conversations. Yeah. There's also a meeting right after the CSS meeting, the day after an FDA analysis data standards, I don't know, something like that meeting in Silver Spring. But you guys, everybody can join remotely. So I encourage you, if you're interested in that, there's a Federal Register notice that came out. You can join that meeting. I'm going to be there for the CDISC meeting, so I'm going to go in person, but anybody can attend that meeting. It's a public meeting, it's free. You just have to sign up and you can listen all day. It's an all-day thing. It's like from 9 to 4 Eastern time. So enjoy being up at 6. Yeah, I did that for the last meeting, and it was the thing at the very end that was of interest, that technical rejection criteria.
So we have officially run, slightly past 4:30, but there's still one thing left to do, and so nobody's going to hold it against you if you have to leave, but do be aware that after we're all done talking here, whoever's left gets to go over to Houlihan's and have some drinks and appetizers, and so that could be fun too.
John's not buying. Neither am I.
So yeah, one of the things we wanted to talk about was, since we do have two vendors or two sponsor companies, Genentech and Gilead, they're going to talk a little bit about a high level and then leave it up for questions around some of the issues with implementation of standards at an enterprise level, some of the governance thoughts, and then sort of how do we achieve efficiencies. So I know Ram talked a little bit about how they're doing that. And so I think once I kind of go through my spiel, we'll kind of pass it off to him to add on. But the first topic, and I'll try to go fast because I know we are over time, was around issues related to.
Speak up a little bit, or you're closer to your mic.
Is this better?
Yes, thank you.
Yeah, that's
okay, perfect. So the first topic was around enterprise level implementation issues and things that, having tried to do many things, at Gilead, what are we finding that is inhibiting us? And so I think the three main topics, and I'm sure it's probably relevant to most companies, is general IT, right? We have a great idea from a business perspective, and there are issues or rules and processes we have to follow from IT, and there's always great reasons and rationale, but we have to go through them oftentimes. And so that's one of the main points to consider, is especially when you start thinking about timelines and, I think an MDR is a great example of, hey, we want to implement an MDR, what's the timeline? I know a lot of vendors will typically say, we can get it up for, 6 months, 9 months. I would just throw out a word of caution. We haven't implemented an MDR. I've spoken to many companies who have, and it tends to be a lot longer than whatever you think it's going to be. There are a lot of steps that you have to go through, and that tends to happen whenever you want to implement an enterprise level software package or system. Security is obviously going to be an issue, especially when a lot of these offerings are going to be cloud-based. If you haven't yet implemented something on the cloud in your company, that's going to be a potential issue. You want to make sure, again, you account for that. You bring in people early on in the process, and make sure you have the right funding internally from business and IT to make sure you can handle, not just the initial costs, but the upkeep and sort of O&M pieces that go into a system or an implementation. And that really is, when I think about it, how do you implement with technology on the standards themselves? Because that's obviously the point, right? You standardize so that you can. But the second piece around change management is, when you implement standards or you implement tools or systems, it's different from what you have done historically. And you have to make sure that, you're constantly thinking about how are we going to make sure we manage the change for every piece of the user, whether it's a direct impact, an indirect impact. And that's been a challenge for us, over the last few years. It will continue to be a challenge because we're implementing things that are forcing people to change how they do things, right? So even if there's a long-term efficiency gain, there's always that short-term hurdle you have to go through. The second piece is around the effective governance. So, I had put out a survey for probably, I think it was about 15 companies to kind of just get a sense of where companies are in terms of standards development, tools implementation, governance. And one of the interesting pieces was a lot of companies are implementing standards, and a lot of companies might have an MDR to store their standards. And as Ron pointed out, the question though is, are my studies following the standards? And is there a system that can be put into place to automate that checking and come up with usable governance metrics or in real time, right, that can actually identify what differences matter? Because you're going to have, and as Ron pointed out, study-specific CRF pages, right, where you're going to have a difference from what would normally be in the standard, which is nothing. Or you'll have a certain question that gets added for a study, and that's going to be a different field and a new field. So you have to make sure that you identify, these are the pieces of our standards that have to be the same. And I would start there, hone the process for how you will govern and adjudicate those differences, reaching out to the programming team or the biostatistician or potentially even clinical research for asking why you're asking for these changes. And then start going down into the sort of medium severity differences. And then eventually going down to all differences, right? And I think automation is going to be key. But at the end of the day, when you have all of that piece, of the puzzle put into place, you still have a report of differences that you'll need to communicate with somebody, right? There has to be that human element. And so I think what people get concerned about is, well, you automate things, and you're going to reduce headcount or you're going to reduce the people that are doing the jobs. That's never going to happen, right? The point is to make the redundant pieces sort of rote and allow the expertise that you have at your companies to do the more complex pieces, right? Or the complex analyses or the complex data set generations. And the goal of that is the efficiencies, right? But when you talk about those efficiencies, I think you always have to have some type of baseline, and that's always going to be a difficult piece to figure out, which is, well, most companies have some type of standard, whether it's a therapeutic area standard, an SDTM light, an Atom light, something that's similar but not necessarily a formalized standard. So when you think about baseline, you always want to figure out, how long did things take before? And I think time is always a good metric, but there's a lot of other metrics that you can use, right? I think Pinnacle 21 came up earlier. I think the number of validations that you find, the validations over time, the time it takes to resolve those findings, right? I think all that are examples of metrics that can point to the effectiveness of standards development and implementation. And there's a lot of other pieces, but I think figuring out then what are those business critical metrics that matter, and then actually going out and collecting those metrics and showing the potential value, right, is now realized based on these sort of critical findings or critical ROI metrics. So that's a very high level. I think we want to leave it up to questions, but Ram, if there's anything you wanted to add, from your side, I think that would be great.
Yep. Thank you, DJ. So from what I see, is like the governance is a big challenge, right? So, and the change management. So we are changing things, and the entire biometrics programming team might have to undergo the change management stuff. So more than we create the tools and do automation and stuff, it is up to the study teams to follow it. They can decide not to use it at all. So there is more of self-assessment from the study teams. So do I need to reinvent the wheel every time, or can I make use of the standards that we already have? So that is a self-assessment that every study team has to make when they are designing a study and try to adhere to the standards as much as possible. That will create a lot of efficiencies down the line.
So if the study team decides not to be compliant, are there sufficient consequences for that failure to persuade them to be compliant the next time?
So if a study team is not adhering to the standards, so obviously their study build time and the time that they take to create the SDTMV and subsequent analysis data sets is going to increase. So from from a developer or from a study team's perspective, it will make sense for them to adhere to the standards. So as long as our standards are comprehensive enough and our supporting tools are comprehensive enough for the team. So I don't think there is any penalizing the study teams for not adhering to the standards. So it's just a good data citizenship culture that we need to embrace and develop within the organization.
Yeah, to add to that, John, I think the real-time component is key, right? Where if you have in real time the ability to assess the compliance or adherence to standards, right, or the deviation, whatever the majority might be, I think that's when you have the ability to have a dedicated team, and it can be a small team, to reach out or to find out, are they playing around with some programming, or are they purposefully deviating, or are they unknowingly deviating, right? I know, again, a lot of people use, Excel-based mapping specs or DDTs. They may accidentally copy a tab from an old study into a new one, and that's where they may see issues, and again, that's where having that real-time assessment, to have someone then go back and say, hey, programmer, I noticed these are the findings. We're not sure why you did that. What's going on? I think once people start seeing that, once we deviate, there's going to be some type of person question asking me. I think that will start, reducing that deviation over time, or at least that's the goal.
Yeah. So one other thing too, I come from Genentech as well. So
we are in the same team.
But I think one of the things Ram mentioned it earlier in his presentation is we're trying to build that individual accountability, and really striving towards having data be fair within Roche Genentech. So I think that builds that accountability onto those individuals. Now, at the end of the day, if your study isn't fair and you're trying to merge it with other content, that's a, that study team is going to be responsible for not using those standards. So I think that's another piece of it. And I think our Shiny app that we have now really highlights where those standards aren't being followed. So I think that helps.
Yep. Thank you, Carla.
Yeah, the struggle that I tend to see, because I work in governance too, is the people who don't want to move to the new standard because their study is so much like this other study. And they just want to of course, when it gets down into it, then their study really isn't that much like the other study at all, and created a bubble of work for themselves. So how do you deal with that? Like that, I don't want to move to the standard because my study is so much like the other.
Yeah, so like we have the FDA rule that we need to be in SDTM 3.2, but like a older study within the same molecule, they have other studies in SDTM 3.13, and they want to do the same, and they'll say that okay, if we go for submission, we'll do SDTM 3.2. So like that is not a fair data citizenship. So you are deviating from the process just for the sake that you can do stuff quicker at that point in time. But in a longer run, it's not going to help the study teams. When it comes for submission, so then you need to undergo the whole exercise of upgrading from 3.13 to 3.2. So it is creating awareness within the study teams, and like explaining them that and like influencing them that like adhering to the latest standards definitely makes sense, rather than like just trying to reuse some of the older programs or some of the older mappings. We should really adhere to the standards and keep the data fair.
Yeah, and I would agree with that. I mean, we used a hard deadline, right? So I mean, I think the 3.2 date, the March 18 and the Define-2 date, I think that was an easy trigger for us. And so part of it was, the studies that were before that, the older studies, we weren't tracking things before. Again, there was this hope that people were following standards, but there was no way of doing it. But after that date, the newer studies, they had to follow the standards. We were able to implement new utilities, new specs that allowed us to do the governance. And when we do get these types of questions, I think it then becomes very easy to sort of upfront say, this is the process. And if there's a real reason to deviate, then we'll want to escalate that. And I think typically, if it's the rationale is simply because I want to use an old program, people typically don't want to escalate up. But if there's some significant reason for it, I think then that's when they start feeling comfortable, and that you can sort of, from a governance perspective, I think you can sort of weed out the people who are doing it for laziness sake, sort of the short-term laziness.
So you're talking about starting up new studies, using these tools and so forth to start up new studies, but individuals who are helping start up new studies, they're also working on old studies at the same time, right?
Yep.
It's a very common scenario.
Yep.
And one of the issues that I've seen sort of crop up again and again is that for an awful lot of people, it's very difficult to sort of keep those two mindsets active, within the workday. And that if you have to switch back and forth between them, that you end up doing a lot of what you would call task switching, right? Which is just putting down one mindset and picking up another one without moving any tiles while you're doing that. What sort of issues do you see along those lines?
Yeah, so we started using this, and we already have a lot of questions like how. So if a new person joins the team, there will be a bunch of studies that follow one process, and there will be a bunch of studies that follows a different process. How do I identify which study follows which process, and how do I know what standards do I follow when I'm working on a study? So we go by the study start date, similar to what we have in the FDA rules. So if a study starts after January 1st, 2019, okay, you have to follow these processes, and these are the tools and the programs, and this is documented in this wiki page. For any studies that has a start date before January 1st, 2019, following these standards, and these documents still exist, but these are not current, that these are not active anymore, but you can still refer for the old studies. You're switching back and forth within the programming environment. If you are working on 2 studies within the same day that follows 2 different processes, it will be challenging for the programmers, but I hope they'll get used to it.
Yeah, I would. The only thing I'd add to that, John, is it's a short-term problem, right? I mean, once they use the new process or the new study spec or the new utility once, now they have a sister study that they can use for the next one, right? So, I mean, we've done testing, and it's always been where the first one takes a little bit longer, but the second one is just on par with the same way they've done things before. So that's sort of the change management piece, which is, hey, if you follow the new stuff, you can use the new cool tools that'll save you time, for other things. So it's just sort of a, you eat a little bit and here's the carrot at the end.
Right, I think we are right on time.
I think, GJ, thanks for staying up so late, DJs.
Hey, of course. No, it's good. I'm able to stay in my room, so.
And thanks you guys for this. To me, it's really interesting to hear how 2 companies with the same problem set or similar problem set, are going at it, and what the commonalities are, the issues that they see. But there's a present possibility that some other group is going to go busting through that door in 5 minutes and go, "This is our room now." So I think we'll have to call it there. Thanks to everyone for having shown up and for showing up on the webinar. In total, we had on the order of 250 or so people, actively participating at the peak. So nice. And a lot of people have asked about whether there's going to be a recording available, and the answer is yes. And we'll also post the presentations. And based on the experience with recordings from previous meetings, we had another 200 people or so view the recordings from the previous meetings. So this is going to get a lot of play. So
thank you so much, John.